to Sleepy Facts with Justin. I'm so glad you're here tonight, wherever you are right now. Whatever kind of day you've had, you can set it all down. You don't need to hold on to any of it. Not tonight. Tonight, we are going somewhere very quiet. Somewhere cool and still and soft. We are going to the forest floor. Close your eyes if they aren't already. Imagine the ground beneath a great old forest. The light here is gentle. It filters down through layers of leaves high above you. And by the time it reaches the forest floor, it has become something softer, something golden and green and slow. The air smells of damp earth, of fallen leaves slowly becoming soil, of something ancient and good. This is one of the quietest places on earth, and yet it is one of the most alive. Beneath your feet, if you were standing there, something extraordinary is happening, something that has been happening for hundreds of millions of years. Long before the first trees grew tall, long before the first birds sang, long before anything with legs ever walked across this earth. The fungi were already here, quietly working, quietly connecting, quietly holding the world together. Tonight, we are going to explore their world. The kingdom of fungi is unlike anything else on earth. Fungi are not plants. They are not animals. They belong to their very own kingdom, a realm of their own making. Sitting gently between the world of the living and the world of what has passed. There are an estimated 3.8 million species of fungi on this planet. Most of them have never been named. Most of them have never been seen. They live in the soil. They grow along roots. They thread themselves through fallen logs and damp leaves and the slow, soft decay of the forest floor. And they do this work silently, patiently, the way the best things in nature always do. Over the next few hours, we will wander gently through this hidden world together. We will follow the white threads of mycelium deep into the soil. We will learn how trees share food and send quiet messages to one another. Through fungal networks so vast and intricate, scientists have called them the Wood Wide Web. We will discover fungi that glow softly in the dark, fungi older than the dinosaurs, fungi that build the soil beneath our feet, one slow season at a time. There is nothing here to worry about, nothing to do, nowhere to be, only the forest floor, stretching out in every direction, cool and dark, and impossibly rich with life. Let your breathing slow down now. Let your body feel heavy and warm. The forest is quiet tonight. The fungi are at work beneath the leaves, and you are exactly where you need to be. Let's begin. You are still here, and that is a wonderful thing. Let the forest settle around you now. Let the sounds of the world outside grow faint and far away. There is nothing urgent here, nothing pressing. Only the soft ground beneath the trees, only the slow, quiet dark. Tonight we begin with a question, a simple, gentle question. What is a fungus? You might think you know the answer. You might picture a mushroom, perhaps a small brown cap pushing up through damp soil, or a cluster of pale white shelves fanning out from the side of a fallen log. And you would not be wrong. But a mushroom is only a small part of the story, only the very tip of something much, much older, and much, much stranger. For a very long time, scientists placed fungi alongside plants. They looked at them and said, yes, these grow from the ground. They do not run or leap or swim. They must be plants. This classification held for centuries, and yet something never quite fit. Plants reach upward toward the light. They gather sunlight and turn it into food. They are, in a sense, engines of light. Fungi do not do this. Fungi do not have chloroplasts. They cannot photosynthesize. They cannot turn sunlight into energy. So if they are not doing what plants do, what are they doing? To understand fungi, you have to let go of the idea that living things must fit neatly into two boxes: the box of plant and the box of animal. Fungi occupy a space that belongs entirely to themselves. They are their own kingdom, a vast and ancient realm, stretching back into a time before we can easily imagine. Scientists now believe the common ancestor of all living fungi existed somewhere between 900 million 1.4 billion years ago. That is a number so large it becomes almost dreamlike, almost peaceful. 1 billion years, long before the first fish, long before the first flower, long before the first breath of anything with lungs. The fungi were already here. In those earliest days, fungi likely lived in the water, the ancient seas, warm and still. Some of those first fungi may have had flagella, tiny, whip-like tails that propelled them slowly through the water. We carry a small piece of that ancient history with us, even now, because molecular science has shown us something remarkable. Fungi are not more closely related to plants. They are more closely related to animals, to us. You and a mushroom share a common ancestor somewhere in the deep, slow ocean of time. This is not a frightening thought. It is a humbling one, a reminder of how connected everything is, how nothing in nature is ever truly separate. The key to understanding a fungus lies in its cell walls. Plant cells are surrounded by cellulose, a soft, papery material that gives plants their structure. But fungal cell walls are made of something different, something called chitin. Chitin is tough and flexible, and here is the beautiful, strange detail. Chitin is the very same material found in the shells of crabs, in the armor of beetles, in the wings of dragonflies. A mushroom and a dragonfly, sharing the same building blocks, a forest floor and a riverbank, quietly connected at the molecular level. This is the way of nature. Everything rhymes. So if fungi are not plants, and they are closer to animals than to plants, how do they feed themselves? Animals eat. They find food and consume it. Fungi do something more subtle, more patient, more gentle. Fungi secrete. They release digestive enzymes into the world around them. These enzymes break down whatever the fungus is growing on. A dead leaf, a rotting log, a grain of soil. And then the fungus simply absorbs the nutrients. It does not chase. It does not hunt. It dissolves and receives. There is something deeply restful about that. Something worth sitting with. The patient art of dissolving and receiving. There are an estimated 3.8 million species of fungi on this planet. Scientists have formally named perhaps 140,000 of them. That means the vast majority of fungi have never been described, never been studied. They live their entire lives unseen, unnamed, quietly working in the soil and the bark and the damp places between. That number, 3.8 million, is almost beyond imagination. If you counted one species every second, it would take you more than 40 days to reach the end, and still, you would only be counting the species we suspect exist. The true number may be higher still. The fungi kingdom contains within it an extraordinary range of forms. There are fungi so small they are visible only under a microscope. Single cells drifting through soil water. And there are fungi so large they challenge our understanding of what a single organism can be. In the Malheur National Forest in the state of Oregon, there lives a honey fungus. Its scientific name is Armillaria ostii, and it is enormous. It covers an area of approximately 10 square kilometers. It weighs an estimated tens of thousands of tons. It may be as many as 8,000 years old. 8,000 years old. While human civilizations rose and fell, while languages were born and forgotten, while rivers shifted course and mountains slowly, invisibly eroded, this one fungus was quietly, patiently growing, spreading one slow thread at a time. Let that image settle over you now. A single organism spreading for 8,000 years, not rushing, not competing, simply growing, thread by thread, season by season, year by year. There is a lesson in that, perhaps. Or maybe it is enough just to rest in the image of it, something ancient and unhurried, reaching gently through the dark earth. The world of fungi is divided into several large groups. Some fungi are tiny and single-celled, like yeasts. Others grow as long chains of cells called hyphae. These hyphae are the fundamental building block of nearly all complex fungi. Each hypha is like a single thread, microscopic and pale almost invisible to the naked eye. And yet, when millions and billions of hyphae weave together, they form something extraordinary. They form mycelium, that dense, white, thread-like mass that you might have seen when you lift a log from the forest floor, the white webbing on the underside, the soft, pale network running through the soil. That is mycelium, and we will spend much more time with it very soon. For now, simply rest with the idea of the kingdom. This vast, ancient, unnamed realm, not plant, not animal, something entirely its own, something that grew in the ancient ocean and learned over millions of years to thread itself through soil, to break down stone, to feed forests, to hold the earth together. Fungi are not dramatic. They do not bloom in bright colors to attract attention. They do not sing or call or move quickly across the ground. They simply work quietly, persistently, and the world is unimaginably richer for it. The forest floor around you is soft tonight. The leaves are damp and still. Somewhere in the darkness, just beneath the surface of the soil, hyphae are growing. Threads finer than spider silk, reaching outward through the dark, breaking down the fallen leaf, absorbing the old, slow carbon, passing it along. There is no sound, no hurry, only the slow, steady work of an ancient kingdom, doing what it has always done, long before we arrived to watch. Let yourself drift now. Let the forest hold you. The fungi are working. Everything is as it should be. You are still here, still resting, still breathing slowly. That is exactly right. Let the weight of your body sink a little deeper into wherever you are lying. Let your shoulders soften. Let your hands be still. The forest is quiet tonight. The air is cool and damp. And beneath the surface of the earth, just a few inches below the layer of fallen leaves and soft, dark soil, something extraordinary is growing. It has been growing all evening. It was growing while you prepared for sleep. It was growing while you closed your eyes. It grows always, quietly and without ceasing. We are going to go deeper now, beneath the surface, into the world that you cannot see. the world of mycelium. Imagine a single thread, finer than a strand of human hair, almost impossibly thin, so thin that dozens of them, laid side by side, would together be no wider than a single eyelash. This thread is a hypha. It is the basic unit of a fungus. Each hypha is a tubular chain of living cells, surrounded by a wall made of that tough, flexible chitin we spoke of earlier. And each hypha grows at its very tip, not from the middle, the way a plant might grow, not from its base, the way a hair grows from a follicle, but from the very front, from the leading edge, reaching forward into the unknown. The tip of a hypha is a restless, searching thing. It pushes through soil particles. It follows gradients of moisture. It reaches toward sources of food, drawn by the faintest chemical signals dissolved in soil water. And as it grows, it branches. One thread becomes two. Two become four. Four become eight. And on, endlessly, in every direction. This branching, weaving, interlacing mass of threads is called mycelium. It is the true body of the fungus, the mushroom you see above ground. That is only the fruit, the flower, the brief, visible expression of something much larger living beneath. The word mycelium comes from the Greek word for mushroom. But mycelium itself looks nothing like a mushroom. It looks like frost on a window pane, or like the tracery of rivers seen from far above, or like the branches of a tree, but impossibly fine, impossibly complex, impossibly beautiful if you could see it clearly. Mycelium is white, most often a soft, pale white, the color of fresh cream or new snow. And when you find it in the forest, beneath a log, threaded through the dark soil, it looks almost luminous, like something that generates its own gentle light. Now consider this number. 1 teaspoon of healthy forest soil. Just 1 small teaspoon contains approximately 10 kilometers of hyphae. 10 kilometers in a teaspoon. Let that be a quiet thought to hold. You could spend the rest of this evening simply sitting with that number. The extraordinary density of life packed into the smallest handful of earth. Every teaspoon of forest soil, a universe of white threads, reaching and branching and growing in the dark. And the forest floor, of course, contains far more than a teaspoon of soil. A single footstep covers hundreds of those teaspoons. A hectare of forest contains billions. Scientists estimate that there are more kilometers of fungal hyphae in the soil of the earth than there are stars in the observable universe, more kilometers of mycelium beneath the ground than we can truly comprehend. All of it growing, all of it working, all of it alive. The mycelium does not simply grow randomly. It is, in a very real sense, intelligent, not intelligent in the way we use that word when we talk about people or animals, but intelligent in the way a river is intelligent, finding the path of least resistance, redirecting when blocked, consolidating where it is rewarded, abandoning paths that offer nothing. When scientists have grown mycelium in laboratory conditions, placing food sources at various points in a dish, the mycelium finds them. It maps the space. It builds efficient routes between resources, the network it creates. The pattern of threads connecting food to food remarkably resembles the networks humans build. Road networks, rail networks, the internet itself, the same elegant logic, arrived at by billions of years of quiet evolution. Through the mycelium, a fungus absorbs the world. The process is simple, though the results are profound. When a hyphae reaches a source of food, a dead leaf, a fragment of wood, a grain of organic matter, it releases enzymes. These enzymes break down the complex molecules of the food source into simpler ones. Sugars, amino acids, minerals dissolved in water, and then these small, simple molecules are absorbed directly through the walls of the hyphae, drawn in, taken up, distributed through the network. This means the mycelium is, at once, a body and a stomach. It digests the world around it as it grows through it. It does not need a mouth or a gut. It does not need to find food and bring it back to a central place. It simply grows toward the food and eats by touching. There is something about this that feels deeply peaceful to consider. A way of being in the world that involves no grasping, no struggle, only slow, patient reaching, only gentle contact, only quiet absorption. The mycelium also does something that scientists are still working to fully understand. It moves nutrients not just into the fungus, but through it. Within the network of hyphae, liquids can flow, drawn by pressure gradients and osmotic forces, like water moving through a series of tiny pipes. This means that something absorbed by one part of the mycelium can travel to a completely different part. Nutrients gathered at the edge of the network can be transported to the center. Resources from one area of the forest can be moved to another. Slowly, quietly, without any apparent effort. This ability to transport nutrients across a network is part of what makes mycelium so important to the forest. And we will explore that much more deeply when we talk about the wood wide web. But for now, simply imagine the flow. A slow, invisible current moving through white threads in the dark soil, carrying the forest's nutrients from place to place. The way a slow river carries sediment, the way a gentle breeze carries seeds. Mycelium is found almost everywhere on land that can support life. Beneath forests, of course, but also in grasslands, in deserts, where it threads through the tiny amounts of available moisture. In tundra, growing slowly beneath the snow, in the soil of your garden, even in the walls of old buildings, quietly breaking down the wood. Wherever there is something to decompose, something to connect, something to feed on, mycelium is likely there, growing at its patient, unhurried pace, one thread at a time. The color of mycelium is most often that soft white, but not always. Some mycelium is pale yellow. Some is faintly blue. Some takes on the color of its substrate, the wood it grows through, the soil it inhabits, and some mycelium glows. In the deep forest, on warm nights, certain fungi produce a faint, cool light, a bioluminescence that has no obvious purpose. That simply is. We will come back to those glowing threads later in their own quiet section. for now, let the image settle gently. A forest at night, darkness all around, and beneath the leaves, the faintest pale glow. White threads, softly luminous, the hidden body of the forest, dreaming in the dark. Mycelium is not permanent. It grows and it dies. When conditions are right, when there is food and warmth and moisture, it reaches outward, expands, branches endlessly. When conditions become difficult, when food runs out or the soil dries, some parts of the mycelium die back. The threads collapse, they decompose, and in doing so, they add nutrients back to the soil. Even in dying, they give. There is one more beautiful thing to hold before we drift a little further into sleep. The mycelium of one fungus can meet the mycelium of another. And when they do, they can recognize one another. If they are the same species and compatible, they may fuse, their threads joining together, their networks becoming one. Two fungi growing toward each other through the dark soil, meeting somewhere in the middle, merging, becoming something larger than either was before. This is how the vast networks of the forest form, thread by thread, joining, connecting, growing outward and inward at once, until the entire forest floor is threaded with a single, vast, interconnected web, a living fabric beneath our feet, holding everything together, quietly, without asking for anything in return. Let your breath slow now. Let the image of the white threads be the last thing in your mind, soft and pale, and reaching in every direction through the dark, growing toward the food, growing toward each other, growing always in the gentle dark beneath the forest floor. You are warm and still. The mycelium is growing. Everything is connected. Everything is well. Still here, still breathing. That is all you need to do tonight. Just breathe. Let the air move in slowly and out again. In the forest around you, the trees are still. Their roots reach deep into the cool earth, and in that darkness beneath the roots, something is moving, something slow and patient and beautiful, something that connects every tree to every other tree. A network so ancient and so vast that scientists, when they first began to understand it, reached for the only comparison they knew. They called it the Wood Wide Web. To understand the Wood Wide Web, we need to understand a word that comes from the Greek, mycorrhiza. It breaks apart into two gentle pieces, myx, meaning fungus, and rhiza, meaning root. Mycorrhiza, then, is simply fungus root, the name for what happens when a fungus and a tree root find each other in the dark soil and form something together that neither could form alone. Mycorrhizal relationships are among the oldest partnerships on Earth. Scientists believe they first formed approximately 500 million years ago. This was around the time the very first land plants were making their tentative journey out of the water, stepping, in a sense, onto dry land for the first time. Those ancient plants had simple, shallow roots. The land was rocky. The soil was thin and mineral poor. And yet the plants survived and thrived, and one reason they were able to do so was the fungi. The fungi were already there, already threading through the rock and soil, already breaking down minerals, already holding moisture. And when the roots of those first land plants reached down, the fungi reached up to meet them. This meeting, this joining, changed everything. It is not an exaggeration to say that the forests of the world, every forest, on every continent, may not exist as we know them without mycorrhizal fungi. Approximately 90% of all land plant species form mycorrhizal relationships. 9 out of every 10 plants, from the tallest redwood to the smallest wildflower, are in partnership with fungi, living not alone, but connected, supported, sustained. There are two main types of mycorrhizal relationship. The first is called ectomycorrhizal. Ecto meaning outside. In this type of partnership, the fungal hyphae wrap themselves around the outside of the tree's root. They form a sheath, a soft, fungal sleeve around each root tip, and then they extend outward from this sheath, deep into the surrounding soil. The fungal threads reach far beyond where the roots themselves could ever go. They explore. they find phosphorus and nitrogen and minerals dissolved in tiny droplets of soil water, and they bring them back to the root. Trees that form ectomycorrhizal relationships include oaks, beeches, pines, and birches. The great trees of the temperate and boreal forests. Cool climate trees. Old world trees. Trees that have been forming these partnerships since before the ice ages. The second type is called arbuscular mycorrhizal. In this partnership, the fungi go further still. They do not merely wrap around the root, they penetrate inside the root cells themselves. Tiny branching structures called arbuscules form within the root cells. Little trees within the trees own cells. These structures are the sites of exchange, the place where nutrients pass from fungus to plant, where sugars pass from plant to fungus. The handshake between two kingdoms happening at the cellular level. And here is the heart of the matter, the heart of what makes this relationship so beautiful, so sustaining, so worth understanding. It is not a one-way exchange. The tree gives to the fungus, and the fungus gives to the tree. The tree, through photosynthesis, produces sugars. It gathers sunlight and carbon dioxide from the air and turns them into sweet, energy-rich carbon compounds. Up to 30% of the sugars a tree produces may flow down through its roots and into the mycorrhizal fungi, and in return, the fungi provide the tree with phosphorus, nitrogen, water, the raw materials of growth, the things the tree cannot easily gather for itself. Two organisms, each with something the other needs, each giving freely, each made stronger by the exchange. But the story does not end there. Because the mycelium of mycorrhizal fungi does not connect one tree to one fungus and stop. It extends outward. It reaches the next tree, and the next, and the next, until the roots of dozens, hundreds, sometimes thousands of individual trees are connected through a single fungal network. A web of threads linking tree to tree across the forest. And through this web, nutrients can flow not just from fungus to tree, but from tree to tree. The scientist who has done more than anyone to to reveal this truth is a Canadian ecologist named Dr. Susan Simard. She spent years studying the forests of British Columbia, watching the trees, wondering how the young seedlings survived in the deep shade beneath the older trees, wondering how carbon moved through the forest, and what she found changed the way we think about forests forever. She discovered that carbon, the sugar carbon of photosynthesis, could travel from one tree to another through the mycorrhizal network. Large, old trees were sending carbon to small, young seedlings. Older birch trees, bathed in full sunlight, were sharing their sugars with young fir seedlings growing in the shade, not competing, not hoarding, sharing. Dr. Simard began to call the largest, oldest, most connected trees by a name that felt immediately true: Mother Trees. These ancient individuals, with their deep roots and vast mycorrhizal connections, sit at the heart of the network. They are linked to hundreds of other trees. Some mother trees have been found to be connected to as many as 47 other trees through a single mycorrhizal network. And they use these connections when a seedling nearby is struggling, growing slowly in the shade, lacking nutrients. Carbon and water and minerals may flow toward it through the network, from the old to the young, from the large to the small, quietly, beneath the soil, without any visible sign. When a tree comes under attack, when insects begin to eat its leaves, or a pathogen invades its bark, something remarkable can happen. Chemical signals travel through the mycorrhizal network. The distressed tree releases compounds into the soil, into the fungal threads, and these signals travel outward through the network to the trees around it. Receiving trees may respond by producing more defensive compounds, preparing their own chemistry before the threat arrives. A warning passed through the web, not in words, not in any language we would recognize, but real nonetheless. Real and effective and ancient. The wood wide web is not the same in every forest. In the cool, temperate forests of North America, Europe, and Asia, where wood decays slowly and organic matter builds up deep and rich in the soil. The ectomycorrhizal networks dominate. These are the vast, highly connected webs, the ones that link tree to tree across great distances. In the warm, wet tropics, where wood decays quickly, different fungi take over. Arbuscular mycorrhizal fungi, forming smaller, more local networks. Each forest has its own version of the web, its own fungal dialect, in a way, its own quiet, underground language. Some of these networks are enormous. A single mycorrhizal network can extend across an entire forest, covering hectares of land, linking trees of different ages, different sizes, different species. All of them part of the same conversation, all of them connected to the same ancient threads. There is a word for what this creates. Scientists sometimes call it a common mycorrhizal network, a network held in common, belonging to no single tree, shaped by all of them together. There is something in that word, common, that carries a warmth, a shared belonging. The forest is not a collection of individual trees competing for space and light and water. It is, in some profound sense, a community, a neighborhood of roots and threads, linked by the patient work of fungi, giving and receiving, season after season, generation after generation. When a tree dies in the forest, the network does not simply forget it. The carbon stored in its roots and trunk begins to move. The mycelium continues to break it down. The nutrients it spent a lifetime gathering are slowly returned to the soil, to the network, to the trees still living. Nothing is wasted, nothing is lost. The old tree feeds the young ones, even in death, through the same threads that connected them in life. Let that settle over you now. The image of the forest, not as a collection of separate things, but as one breathing, connected, living whole. The trees above, the roots below, and between them the fungi, threading everything together, holding the whole world in a net of white and silver threads. You are resting now. Your breathing is slow, somewhere in the dark, beneath the forest floor. Nutrients are moving. Carbon is flowing. Old trees are giving to young ones. Everything is connected. Everything is cared for. Everything is well. You are drifting now, gently, the way a leaf drifts on a slow current of water, not going anywhere in particular, simply moving, simply resting in the flow. The forest is all around you tonight. Above you, the canopy is still, the branches barely move, and beneath you, deep in the cool, dark soil, conversations are happening, not in words, not in sounds you could ever hear, but real nonetheless, patient, ongoing. The quiet language of trees. We tend to think of trees as silent things, as still things. We see them standing in the forest, and we think, here is something that does not move, does not speak, does not reach out to the world around it. But this is only because we are not listening in the right way. We are listening for the wrong kind of sound. Trees do not speak in the air. They speak in the soil. And they speak in a language made not of sound, but of chemistry, of molecules, of slow electrical pulses moving through living tissue, of signals so subtle that it took scientists many decades to even recognize them. The first and most ancient language of the trees is the one that flows through the mycorrhizal network. We spoke of this network in our last section. The vast web of fungal threads connecting tree to tree through the soil. And we know that nutrients flow through this network. Carbon and phosphorus and nitrogen moving from tree to tree, but nutrients are not the only thing that flows. Signals flow too. Chemical messages, hormonal whispers, slow, faint electrical pulses, traveling through the fungal threads from one tree to the next, from one part of the forest to another. Imagine a tree at the edge of the forest, a birch perhaps, its leaves trembling in the late afternoon light. And now imagine that a caterpillar begins to eat those leaves. One small leaf, then another. The tree feels this, not the way you or I feel a pinch. Not with pain, not with awareness, but chemically. At the cellular level, something changes. The damaged leaf cells release a signal, a hormonal compound called jasmonic acid begins to spread through the tree. This compound acts like an alarm. It tells the tree's other cells to begin producing defensive chemicals. Bitter tannins, compounds that make the leaves harder to digest. Substances that deter the feeding insect. The tree is, in its own quiet way, defending itself. But here is where it becomes extraordinary. The tree does not keep this alarm to itself. The signal travels down through the roots. It enters the mycorrhizal network, and it flows outward through the fungal threads to the roots of nearby trees. The neighboring trees receive the signal, and they respond before any caterpillar has reached their own leaves, before any threat has arrived. They begin to produce their own defensive compounds, preparing, quietly readying themselves. A warning passed through the web. Scientists who study this process have called it interplant signaling through mycorrhizal networks. It is a formal and careful phrase for something that feels, in its essence, quite simple. A community looking out for itself, a neighborhood of trees connected through the soil, sharing information about threats, sharing the knowledge of danger, so that all may be a little safer. But the language of the forest does not only travel underground. There is a second kind of conversation happening in the open air, above the roots, above the soil, in the space between the trees. Trees release chemicals into the air called volatile organic compounds. Scientists shorten this to VOX. The word volatile simply means they evaporate easily. They drift. They disperse on the breeze, and they carry information. When a tree is under stress, when it is being eaten, or when it is short of water, or when disease begins to affect its bark, it releases specific combinations of these airborne compounds. Different stresses produce different chemical signatures. A tree being eaten by caterpillars releases different volatiles than a tree suffering from drought, and the trees nearby can detect these compounds. They absorb them through their leaves, and they respond to what they detect, activating defenses, adjusting their chemistry, preparing for what may be coming. Scientists demonstrated this beautifully in a study of beech trees in Natural natural forest conditions. They tracked the flow of volatile compounds from a source tree, a tree that had been damaged by insects, and they found that the beech trees growing downwind of the damaged tree, in the direction the breeze was blowing, accumulated higher levels of salicylic acid in their tissues. Salicylic acid is a key defensive compound, a natural version of what we find in aspirin, and the trees downwind had more of it. They were more defended, less damaged by pathogens and herbivores. The upwind trees, by contrast, showed no such change. They had not received the signal. They had not had the chance to hear the message carried on the air. Consider what this means. A forest after a storm. One tree damaged by a falling branch. Its leaves torn. Its bark scraped. And in response, invisible compounds drifting outward on the morning air, absorbed by the leaves of the trees nearby, triggering quiet chemical responses throughout the neighborhood. All of it invisible, all of it silent to our ears, and all of it happening with a slowness, a gentleness, a patience that is deeply calming to contemplate. There is a third form of tree communication that scientists are only beginning to understand. Electrical signals, not loud or dramatic electrical signals, not the crack of lightning or the spark of a wire, but slow, faint, pulsing electrical signals moving through living tissue, traveling through root networks, carrying information from one part of a tree to another. And perhaps, some scientists believe, from one tree to another through the mycorrhizal connections. These signals are so faint, they require extremely sensitive instruments to detect. They move at a pace that seems almost impossibly slow. Centimeters per hour in some cases. Not rushing, not urgent, simply moving. The way a thought sometimes moves through a half-sleeping mind. Slow and soft and barely there when we put all of this together. The underground chemical signals, the airborne volatile compounds, the faint electrical pulses. We begin to see the forest in a new way, not as a collection of isolated organisms, each struggling alone for light and water and space, but as a community, a deeply connected, richly communicating community, where information flows in multiple directions at once, where the well-being of one affects the well-being of others, where the old support the young, where warnings are shared freely, where nothing is kept private when sharing would help. There is something here that asks us to soften the boundary between what we call intelligent and what we call non-intelligent, between what we call communication and what we call mere chemistry. Is it communication if there is no intention? Is it language if there is no mind behind it? These are questions scientists continue to discuss with great care, with appropriate caution, and yet the effect is undeniable. The signals pass, the trees respond, the forest is healthier for it, whatever we choose to call it. Dr. Susan Simard, whose work we touched on in our last section, has spent her career sitting with these questions, watching the forests of British Columbia, counting the connections, measuring the flows, and she has said something beautiful. She has said that the forest is not a collection of trees. It is a community of trees, and that communities have memory. The old trees carry within their vast mycorrhizal connections a kind of history of the forest, a record of past stresses, past partnerships, past losses. The forest remembers. Let that be a gentle thought to carry into sleep. The forest remembers, not the way we remember, not in images or words or feelings, but in chemistry, in the slow, patient record kept by living threads in the dark soil. The forest has been here longer than we have. It has survived drought and fire and the slow creep of ice. And through all of it, the trees have been passing messages. Through the soil, through the air, through the ancient networks that connect them. Quietly, without ceasing, you are part of a world that is far more connected than it appears. Every forest you have ever walked through, every tree you have ever touched, was speaking in its own quiet way, sending and receiving, passing information through the ground, through the air, through the patient, invisible threads of the fungi that hold the forest together. Tonight, as you drift toward sleep, let that connection settle over you You are not separate from the world. You never were. You are part of the same vast, breathing, quietly communicating whole. Let your thoughts slow now. Let them drift like compounds on a gentle breeze, going nowhere in particular, simply drifting, simply dissolving into the soft, connected dark. You are still here, still breathing slowly. Good. There is nowhere else to be tonight. Only this. Only the soft dark. Only the forest, quiet and still around you. Let your body be heavy. Let your thoughts be loose and unhurried, and let me tell you about something that happens every single moment of every single day in every forest on earth. Something so ordinary it is invisible, and so important that without it, life as we know it could not exist. The art of decomposition. When a leaf falls from a tree, it does not disappear. When a branch breaks and tumbles to the forest floor, it does not simply vanish. When a great old tree finally falls after decades or centuries of standing, its trunk does not dissolve overnight, but it does, slowly and quietly and completely, return to the soil, return to the elements it was made from, return to the great slow cycle of matter that has been turning since before the first tree ever grew. And the ones who do this returning, the ones who make it possible, are the fungi. There is a word for fungi that feed on dead and decaying matter. Saprotrophic. It comes from the Greek sapros, meaning rotten, and troph, meaning nourishment. Rotten nourishment. It sounds, at first, unpleasant, but hold it gently for a moment. Rotten nourishment. The idea that decay itself is nourishing. That what has died becomes, through the patient work of fungi, the foundation of new life. There is something deeply reassuring about that, something almost beautiful. Nothing wasted, nothing truly lost, only transformed. Saprotrophic fungi are the forest's primary recyclers. They are the main agents responsible for breaking down the vast quantities of organic matter that fall to the forest floor each year. In a temperate forest, a single tree may shed tens of thousands of leaves each autumn. Multiply that by every tree in the forest. Add the fallen branches, the dead bark, the shed insect casings, the bodies of small animals, the accumulated duff of decades. Without something to break all of this down, the forest floor would eventually be buried under an ever-deepening layer of its own debris. The nutrients locked inside the leaves and wood would be unavailable. The soil would become thin. The forest would slowly starve. Instead, the fungi go to work. The process begins almost the moment a leaf touches the ground. Fungal spores are everywhere in the forest air. They land on the fallen leaf. They sense the moisture, the nutrients, the chemistry of the leaf surface, and they begin to germinate. Hyphae emerge, tiny threads, pale and fine, beginning to spread across the surface of the leaf, releasing enzymes. These enzymes are the tools of decomposition. Different enzymes for different molecules. Cellulases, which break down cellulose, the tough structural fibers of the plant cell wall. Hemicellulases, for the hemicellulose matrix that holds the cellulose together. Pectinases, for the pectin compounds in the cell walls. Each enzyme a tiny, patient tool, undoing molecule by molecule what the tree spent years building. As the enzymes do their work, the complex molecules of the leaf break apart. Long chains of cellulose become shorter. Shorter chains become simple sugars. Simple sugars are absorbed through the walls of the hyphae, used by the fungus for energy, for growth, for spreading further through the leaf. And as the fungus extracts what it needs, it releases the rest back into the soil. Carbon dioxide breathed back into the air. Nitrogen, phosphorus, and minerals dissolved into the soil water, becoming available once more for the roots of living trees, for the mycelium of other fungi, for the bacteria and the mites and the springtails, and all the other small, hidden creatures of the forest floor. This is nutrient cycling, the great slow wheel of matter turning through the ecosystem. Nothing created, nothing destroyed, only moved, only transformed, only passed from one living thing to another in an endless, patient relay. The fungi are at the heart of it. Without their enzymes, the wheel slows. Nutrients stay locked in dead matter. New life cannot grow. With their enzymes, the wheel turns smoothly. Death becomes soil. Soil becomes plant. Plant becomes leaf. Leaf falls, and the fungi begin again. Wood is a more difficult challenge than a leaf. A leaf is relatively soft. Its cells are thin-walled. Its compounds break down with comparative ease. But wood is different. Wood is tough. It is dense. It is built to last. And the reason it is so tough is a molecule called lignin. Lignin is one of the most complex organic polymers in nature. It is the substance that gives wood its rigidity, its strength, its resistance to compression and bending and breaking. A standing tree relies on lignin, the way a building relies on its steel frame. And lignin is extraordinarily difficult to break down. Most organisms cannot break down lignin at all. Bacteria struggle with it. Simple chemical processes are too slow. But certain fungi have evolved enzymes capable of doing what almost nothing else can. Breaking apart the complex, three-dimensional structure of lignin, unraveling it, dissolving it. These fungi are the only organisms on Earth capable of fully decomposing wood. Without them, the forests of the world would be very different places. Fallen trees would persist for centuries. The forest floor would fill with impenetrable woody debris. Nutrients would remain locked away. Instead, the wood-rotting fungi work their patient alchemy, and the fallen tree is returned to the earth. This process does not happen quickly. A fallen log in a cool, temperate forest may take many decades to fully decompose. During that time, it passes through a series of stages. In the early stages, only the softest tissues are affected. The bark begins to loosen, the outer wood softens. Fungi thread through the outermost layers. In the middle stages, the log becomes home to an entire community. Beetles and centipedes and millipedes, mosses and liverworts on the outside, fungi of many species within, some breaking down cellulose, others specializing in lignin. A whole, quiet ecosystem living inside the fallen tree. In the final stages, the log has become soft throughout. It crumbles gently at the touch. It has given up most of its nutrients to the soil, and it has become itself a kind of soil, dark and rich and teeming with microscopic life. Foresters have a name for this last stage. They call it nurse log, a fallen tree so fully decomposed, so transformed, that seedlings can take root in it. Young fir trees growing from the softened remains of old ones, their roots wrapped around what once was a trunk, reaching through the decomposed wood into the soil below. The old tree, in its final act, becoming the cradle for the new. There is something profoundly moving about this, something that asks us to think differently about endings. The rate of decomposition varies across different forests. In the warm, wet tropics, fungi are incredibly active. The heat and the moisture accelerate their growth. A fallen leaf may decompose within weeks, a fallen branch within months. The turnover is rapid. The nutrients cycle quickly back into the living forest. In cool or dry climates, decomposition slows. A log in a boreal forest, where winters are long and cold, may persist for a century or more. The fungi work more slowly in the cold, but they work, patient and persistent, through the frozen months, through the dry summers, always working, never fully stopping. One more thing to hold gently before we drift further. When fungi decompose organic matter, they do not simply release everything back into the soil. Some of what they process becomes incorporated into their own bodies, into the hyphae themselves. And when those hyphae eventually die, and hyphae do die constantly, even as the network grows in other directions, the materials within them are released slowly into the soil, building up the organic matter, contributing to the dark, rich humus that makes forest soil so fertile, so moisture retaining, so alive. The fungi are not only recyclers, they are builders, builders of the very ground we walk on, patient constructors of the world's richest and most complex substance, living soil. The forest floor is working tonight. As you breathe slowly in the dark, out there somewhere a leaf is being undone. A fallen branch is slowly softening. A nurse log is crumbling, grain by grain, back into the earth. The fungi are at work, enzymes dissolving, hyphae absorbing, nutrients releasing. The great slow wheel turning, nothing wasted, nothing lost, only the long, patient, beautiful work of transformation. Let yourself transform tonight, too. Let the day dissolve. Let what is heavy and complex break down into something simpler, something softer, something that can return gently to the earth. You are safe. The forest is working. Everything returns. Settle a little deeper now. Let whatever tension remains in your body simply soften. Your shoulders, your hands, the space behind your eyes. Let it all be soft. You have been listening for a while now, and perhaps some part of you has already drifted. That is perfectly fine. Let whatever part of you needs to sleep, sleep. The rest of you can stay here, gently, in the forest, where the air is cool, and the fallen logs are everywhere, pale and dark, and slowly, quietly changing. Tonight, we are going to look very closely at a fallen log, not from above, not from a distance. We are going to look at what is happening inside it, in the wood itself, in the slowly transforming grain and cell and fiber of a tree that has fallen and given itself back to the forest. Pick a log in your mind. Perhaps it is a large one, an old beech or a fallen oak, thick as a doorway, lying across the leaf litter. Its bark is still mostly intact on the outside. Mosses have begun to creep across its surface. A small fern has taken root in a crack near one end, but inside the wood in the dark, between the close-packed cells of that once living trunk, something is happening. Something has been happening for years. The fungi have arrived, and they are choosing very specifically what to eat. We spoke in our last section about lignin, that extraordinarily tough, complex molecule that gives wood its strength, its rigidity, its resistance. And we said that certain fungi have evolved the ability to break it down, but not all fungi approach wood in the same way. Not all of them eat the same parts. And the difference between the two main strategies, two ancient, elegant approaches to the problem of wood, produces two very different results, two different kinds of decay, two different textures, two different colors. We call them brown rot and white rot. Let us begin with brown rot. Brown rot fungi are ancient. They are among the most common wood decomposing fungi in the cool coniferous forests of the north. In the boreal forests of Canada and Scandinavia and Siberia, where pine and spruce dominate, brown rot fungi are the primary agents of decay, and their strategy is a precise and particular one. They do not eat everything. They eat the cellulose, the hemicellulose, the carbohydrate scaffolding of the wood cell wall. They leave the lignin alone, or rather, they modify it slightly, oxidize it, change its chemistry. But they do not fully break it down. They leave it behind, and the lignin, stripped of the cellulose that surrounded it, changes color. It turns brown, a warm, reddish brown, the color of autumn leaves, the color of old, dry earth. And then something else happens. As the cellulose is consumed and the lignin is left behind, the wood begins to lose its structural integrity. It becomes brittle. It loses its flexibility, and it begins to crack. Not in long, splintering cracks along the grain, but in a very specific and almost geometric pattern. The wood cracks both along the grain and across it, creating small, roughly cubical pieces, blocks of brown, crumbling wood. This pattern, this cubical cracking, is so distinctive that it gives brown rot one of its other names. Cubical rot. If you have ever walked through an old forest and noticed a fallen log that crumbled into small brown cubes when you touched it, that was brown rot. That was the patient work of fungi, consuming the cellulose, leaving the lignin, leaving the wood to crack and crumble in its characteristic way. And those brown, cubical crumbs are not waste. They are extraordinarily valuable. Brown rotted wood absorbs Lignin-rich residue holds moisture in the forest soil. It builds water retention. It prevents the soil from drying out in summer. It is, in its own quiet way, a gift. The brown rot fungi consume what they need, and what they leave behind sustains the forest in a different way. Now let us turn to white rot. White rot fungi have a different strategy, a more ambitious one in some respects, a more complete one. White rot fungi can break down not just cellulose and hemicellulose, but lignin as well. They possess a suite of remarkable enzymes. Enzymes called lacases, peroxidases, manganese peroxidases. These enzymes are some of the most powerful biological catalysts known to science. They attack the complex structure of lignin directly, unraveling its interlocking rings of carbon, breaking it apart into simpler compounds, releasing its stored energy and matter back into the ecosystem. The result of white rot is very different from the cubical crumbling of brown rot. When white rot fungi consume wood, they remove both cellulose and lignin. What is left behind is pale, often white or cream-colored or faintly yellow. The wood becomes stringy, fibrous. It separates into soft, pale strands, almost like wet paper or old fabric that has lost its weave. You can pull it apart between your fingers. It has no rigidity left, no resistance. All the tough architecture of the wood dissolved, returned to its simplest elements. White rot fungi include some of the most familiar mushrooms in the forest. The oyster mushroom, those beautiful, fan-shaped fruiting bodies that grow in pale clusters from the sides of fallen logs and dying trees. They are white rot fungi, working from within the wood, consuming both cellulose and lignin, leaving behind that pale, soft, fibrous residue, and producing in the process some of the most recognizable and elegant mushrooms in the temperate forest. There is a remarkable piece of history woven into the story of white rot fungi, a piece that connects the forest floor to the coal, seems deep beneath the ground, to the energy that powers cities, to the long buried record of of ancient life. 300 million years ago, in the period we call the Carboniferous, the age of coal, something was different about the world. The forests of that ancient time were vast. Enormous trees grew in tropical swamps. They fell, and when they fell, they did not fully decompose. The lignin in their wood was extraordinarily resistant, and the white rot fungi had not yet evolved the full suite of enzymes needed to break it down. So the wood accumulated, layer upon layer, millennia upon millennia, compressed by the weight of rock, transformed over vast spans of time into coal. The coal deposits that lie beneath the continents today, the coal that humans have been burning for centuries, is in large part the undecomposed wood of ancient forests, wood that the fungi of that time could not fully process. And then, approximately 290 million years ago, something changed. A lineage of fungi evolved new enzymes. The lignin-degrading enzymes of the white rot fungi. And with those enzymes, the great accumulation of wood slowed. The coal-making slowed. The fallen trees began to decompose more completely. The carbon that had been locked away in vast underground seams began to cycle through living systems once more. An evolutionary moment so significant that it changed the chemistry of the atmosphere, that it changed the course of life on Earth. And it happened quietly. In the wood, in the fungi, in the slow, patient work of decomposition. There is also a third type of rot, less commonly discussed. Scientists call it soft rot. Soft rot fungi work more slowly than either brown rot or white rot fungi. They tend to specialize in the outermost layers of wood, particularly in environments that are very wet or very dry or very cold, places where the other fungi struggle. Soft rot leaves wood with a dark, softened surface, often with small cavities just beneath the outer layer. A honeycomb of tiny spaces, excavated by patient hyphae. It is the slowest of the three strategies, but it reaches places the others cannot. Into wet wood at the edge of streams, into fence posts standing half in water, into the compressed, cold soil of northern forests, always working, always patient, brown and white and soft. 3 approaches to the same great task. 3 quiet strategies evolved over hundreds of millions of years, each perfectly suited to a different type of wood, a different environment, a different set of conditions. Together, they ensure that almost no wood escapes decomposition. Almost nothing goes unprocessed. The forest floor is covered in fallen logs in every stage of this transformation. From the freshly fallen, still firm and pale inside, to the cubical brown crumbles, to the pale, stringy fibers, to the dark, barely there softness of the final stages. All of it moving, all of it becoming, all of it on its way back to the soil. The log in your mind is soft now. The mosses on its surface are bright and green. A beetle has made its home in a gap between bark and wood. Inside, the white rot fungi are working. Pale threads through pale, softening wood. The lignin unraveling, the carbon releasing. Quietly, without hurry, without drama. Just the slow, patient, ancient art of transformation. You are part of that same slow world tonight. Let yourself soften. Let the edges of the day dissolve. Let the fungi do their work. Everything returns. Everything becomes something new. Rest now. The forest is taking care of itself. Still here. Still breathing. Good. Let the forest settle around you a little more. Let the sounds of this section carry you somewhere even quieter, even stiller. Tonight, we are going to talk about something very small, something almost impossibly small. Something that drifts on the air around you right now, tonight, wherever you are in the world. Something ancient and patient and full of possibility. We are going to talk about spores. Close your eyes a little more tightly for just a moment. Picture the most complete darkness you can imagine. Now, let a single point of pale light appear. So small, it is barely a point. The size of a dust grain. The size of a thought. That is a spore. The seed of the fungal world. The beginning of every mycelium, the the starting point of every mushroom you have ever seen. A single cell wrapped in a coat of extraordinary toughness, waiting for the right moment to begin. Spores are the way fungi travel. Mycelium, for all its extraordinary reach, is anchored. It grows outward from a fixed point. It cannot pick itself up and move to a new forest, a new log, a new patch of soil on the other side of a mountain, but a spore can. A spore weighs almost nothing. It is a single cell, often just a few thousandths of a millimeter across, so light that the slightest movement of air can lift it, and once lifted, it can travel. Airborne dispersal is the most common way fungal spores move through the world. The wind picks them up from the surface of a mushroom or a bracket fungus or a puffball and carries them upward, sometimes into the airstreams above the forest canopy, into the upper atmosphere. Scientists have found fungal spores in air samples taken at extraordinary altitudes in the lower stratosphere, kilometers above the surface of the earth, and some of those spores, retrieved from those great heights, were still alive, still viable, still capable of germinating if they landed somewhere suitable. A spore drifting in the stratosphere, thousands of meters above the ground, moving with the great slow currents of the upper atmosphere, crossing oceans, perhaps, mountain ranges, entire continents, carrying in its single cell the potential for an entire fungal network, an entire forest relationship, an entire ecosystem of connection, in something smaller than a grain of salt. The spores we are most familiar with are the ones produced by mushrooms. And the way a mushroom produces and releases its spores is a quiet marvel of design. When we look at the underside of a mushroom cap, we see the gills, those thin, blade-like structures radiating outward from the stem, pale and delicate, almost like the pages of a tiny book. And on the surfaces of those gills, billions of microscopic structures called basidia are lined up in rows. Each basidium is a small, club-shaped cell, and at the tip of each basidium, spores develop, usually 4 of them per perched on tiny pointed stalks called sterigmata. Like 4 small drops of water balanced on 4 fine needles. When the spores are ripe, the process of release begins. A tiny droplet of fluid appears at the base of each spore. This droplet changes the surface tension around the spore, and in a fraction of a second, the spore is launched, fired from its basidium, moving at approximately 8 meters per second. A tiny projectile hurled outward into the space between the gills, and then caught by the air, drifting downward, out from beneath the cap. Into the world, a single mushroom can release millions of spores per hour. Some large mushrooms release billions over the course of a few days. The giant puffball, that pale, football-sized fungus, sometimes found in fields and forest edges, is perhaps the most spectacular spore producer. A large giant puffball can contain several trillion spores within its white, papery interior. Trillion. When it is ripe, it cracks open, and any disturbance, a footstep, a gust of wind, a raindrop, sends a puff of spores into the air. A small cloud of pale, microscopic possibility, drifting outward from that single white sphere, into the forest, into the air, into the world. Most of those trillions of spores will never germinate. They will land in the wrong place, in water, or on bare rock, or in soil that lacks the right conditions. They will dry out, be eaten by insects, drift too far from any suitable food source. The odds are extraordinary. The chances for any single spore vanishingly small, and yet the system works. It has worked for hundreds of millions of years because the numbers are so vast that even the tiniest fraction of successes is enough. Even one spore in a trillion, landing in exactly the right place, with exactly the right conditions, is enough to begin again. When a spore does land somewhere suitable, something begins. The spore senses moisture. It senses the chemistry of the environment around it, the temperature, the presence of certain compounds in the soil. And if everything is right, if the conditions align, the spore breaks its dormancy. A tiny crack forms in its outer wall. A single hypha emerges, the very first thread of what may become a vast mycelial network, pushing gently out into the dark soil, beginning to grow, beginning to branch, beginning to reach, all from that single dust grain-sized starting point. Not all spores travel by air. Some travel by water, carried in streams and rain, washing through the forest floor, moving from one part of the soil to another, dispersing through the microscopic rivers of water that move between soil particles after rain. Others travel by animals. When a squirrel or a vole or a chipmunk eats a mushroom, it consumes spores along with the flesh. Those spores pass through the animal's digestive system, and when the animal deposits them elsewhere in the forest, they arrive in a new location, encased in a small packet of organic material, a ready-made bed of nutrition, a gift of sorts, from the animal to the fungi, and from the fungi eventually back to the forest. There is a beautiful relationship between mycorrhizal fungi and the animals of the forest. Truffles, those extraordinary underground fungi that we will spend more time with in a later section, do not release their spores into the air. They cannot. They grow completely underground. They rely entirely on animals to find them, to dig them up, to eat them, to carry their spores to new places in the forest. This is why truffles smell the way they do. That rich, deep, extraordinary scent. It is a signal, a broadcast to the animals of the forest. I am here. I am ripe. Come and find me. The truffle cannot travel, so it invites the world to come to it, and the world obliges. There is one more aspect of spores worth holding gently as we drift toward deeper sleep. The resilience of spores is remarkable. A spore is built to survive, wrapped in a coat of chitin and other complex compounds, resistant to heat, to cold, to desiccation, to ultraviolet radiation. Spores have been found in environments that would kill most living things, in soils frozen for years, in conditions of extreme aridity, in volcanic soils still warm from eruption, and when conditions finally improve when moisture arrives and warmth and the right chemistry. They wake, patient beyond measure, waiting without urgency, ready to begin whenever the moment is right. There is something deeply soothing in that image, a spore drifting on the quiet air of a forest night, weightless, unhurried, carrying everything it needs within a single, impossibly small cell, ready to become something vast and connected and alive. When the time is right, when the conditions align, not forcing anything, not rushing, simply drifting, simply waiting, simply being. Let yourself be like that tonight, not forcing sleep, not trying to stop thinking, simply drifting, letting the currents of the night carry you where they will. You are light enough to drift. You are patient enough to wait. The right moment will come. It always does. The forest air is full of spores tonight, millions of them, drifting on the cool dark, each one carrying within its tiny coat of chitin the possibility of something new. Let that thought carry you gently forward into the soft, still dark. You are doing so well tonight, still here, still breathing slowly. The forest is dark and still around you. The air has a coolness to it now, the kind of coolness that only comes in the deep of night. And out in the forest, somewhere in the field at the edge of the trees, something is waiting to be found, something you would need to stand back to see properly, something that only reveals its full shape from a little distance. A circle, a perfect, patient, ancient circle in the grass. Fairy rings. Even the name is soft. Even saying it quietly to yourself slows something down. Fairy rings. Circles of mushrooms appearing overnight in fields and forest clearings, appearing seemingly from nowhere, returning year after year to the same place, growing a little larger each season, quietly, without explanation, until you know what to look for. For most of human history, the circles were a mystery, and when something is beautiful and mysterious and appears overnight in a field, the human mind reaches for stories, for explanation woven from wonder. The folklore of fairy rings stretches back at least to the 12th century in Europe, across England, Scotland, Wales, France, Scandinavia, and beyond. Cultures looked at these circles and saw something beyond the natural, something magical, something belonging to a world just beside our own. In English and Celtic traditions, fairy rings were said to be the dancing places of fairies. The little people came out at night in these circles and danced round and round, their feet wearing the ring into the grass. The mushrooms were their seats, their tables, their resting places when the dancing slowed. And any human who stumbled into the ring, who crossed that circle of mushrooms in the dark, was in danger. They might be forced to join the dance, round and round with the fairies, unable to stop, unable to leave, dancing until they went mad or fell or wasted away with exhaustion. The rings were places of enchantment, beautiful and treacherous, not to be entered lightly. In Wales, the stories softened a little. The rings were said to mark the location of underground fairy villages. The mushrooms above were only the tops of tiny chimneys or doors opening upward into a world below the roots, and the land around the rings, it was said, was fertile and lucky. Crops grown nearby would flourish. Livestock feeding in fields with fairy rings would be healthy and content. In France, the rings were guarded by giant toads with glowing eyes. In the Netherlands, they marked the place where the devil had set down his milk churn. In Austria, they were burned into the earth by the tail of a flying dragon. Every culture, its own story. Every culture reaching for the same wonder. The scientific explanation arrived gradually, and it is, in its own way, no less remarkable than the folklore. A fairy ring begins with a single spore. One spore landing on the soil, germinating, sending out its first hypha, then branching, growing outward in all directions from that central point, equally in every direction, at a roughly equal rate. And because the growth is equal in all directions, the mycelium forms a circle, a perfect, expanding circle. At the front edge of the circle, the growing edge, the mycelium is at its most active, most productive, most hungry. As the mycelium grows outward and consumes the organic matter in the soil, something happens to the grass or vegetation above. The active front of the mycelium releases nitrogen as it breaks down organic matter. This nitrogen feeds the grass, and the grass responds. It grows thicker, greener, more lush than the grass around it. This is the green ring you sometimes see, the bright, vivid circle of extra green grass, the visible trace of the mycelial front below. Just behind this front, the mycelium is denser, and sometimes this density prevents water from reaching the soil below. The grass here may struggle, may thin out, may die in a band, creating in some rings a pattern of alternating zones, a green outer ring, a dead or thinning inner ring, and then further in still, where the old mycelium has died and released its nutrients back into the soil, a second zone of lush growth. Each year, a fairy ring grows a little larger. The mycelium advances outward. The ring expands. The diameter increases. And by measuring the diameter, by knowing roughly how fast mycelium grows, it becomes possible to estimate the age of a ring. The growth rate varies by species and conditions, but mycelium typically advances by roughly 15 to 45 centimeters per year. A ring with a diameter of 10 meters might be 50 years old. A ring of 50 meters, 2 or 3 centuries. And the great rings, the truly ancient ones, near the city of Belfort in northeastern France, there is a fairy ring. It was formed by a fungus called Infundibulicide geotropa. The fairy ring champignon. Its diameter is approximately 600 meters, nearly half a mile across, and based on the growth rate of its species, scientists estimate that this ring is around 700 years old. 700 years. This ring was growing when the great cathedrals of Europe were being built, when plague moved across the continent, when the maps of the world were still full of blank spaces. All that time, quietly, patiently expanding, one season at a time, one small increment of growth per year until it became something you could almost see from the sky. Aerial photographs have revealed fairy rings much larger still. Rings up to 200 meters in diameter have been photographed from the air, their green and brown and pale circles visible as gentle curves in the landscape. Curves so large they are invisible from the ground. Only from above do they reveal their shape. Ancient patterns pressed into the land by patient fungi, quiet circles, growing for centuries without anyone noticing. There are around 60 species of fungi that can form fairy rings. Some are saprotrophic, the free rings growing in open grassland, feeding on thatch and buried organic matter. Others are mycorrhizal, the tethered rings growing in woodland, connected to the roots of trees. The most famous fairy ring mushroom is called Marasmius oreads, a small, delicate mushroom with a pale tan cap. It appears in rings in lawns and meadows after rain. It is edible and has a pleasant, mild flavor, and it is extraordinarily resilient. When dried completely, when all moisture has left its cap and it has shriveled to a thin, papery disc, it does not die. Give it moisture again, and it rehydrates. It returns to its original shape. It produces new spores. It begins again. There is no fanfare in this, no drama, simply persistence, simply the quiet refusal to be finished. The rings in woodland are different in character from those in open fields. In a forest, the fairy ring may not be visible in the soil and grass at all. No green circle, no dead band. Only in autumn, a sudden appearance of mushrooms pushing up through the leaf litter. In a ring, sometimes a complete ring, sometimes a partial arc where the soil conditions vary. These woodland rings are often the rings of mycorrhizal fungi, connected to the roots of the trees above, part of the vast network we have already explored. Their circles are as ancient as the trees they accompany, and they are invisible for most of the year, patiently working in the dark, revealing themselves only briefly in the fruiting season, a quiet announcement. I am here. I have always been here. Perhaps the most beautiful thing about fairy rings is this: they are a shape that nature returns to again and again. The circle, not because any intelligence chose it, not because any plan was made, but because even growth in all directions naturally produces a circle, the most efficient shape, the most elegant, the shape that has no beginning and no end. Tonight, somewhere in a meadow, somewhere at the edge of a forest, a fairy ring is growing, a millimeter or two outward in the dark, too slowly to see, too quietly to hear, but real, patient, ancient. A circle that began with a single spore in some other century and has been growing slowly and without ceasing ever since. Let that circle hold you tonight. Let yourself rest inside it. Nothing can enter unless you choose. Nothing can hurry you. You are inside an ancient ring, safe, still, held in a circle that has been growing for longer than you can imagine. Rest now. You are drifting beautifully now. There is no need to hold on to anything. No need to follow every word. Let the sound of this voice be something like rain on a roof, something soft and steady and barely there. If your thoughts wander, let them wander. If sleep is beginning to come, let it come. You are safe. The forest is still around you. And tonight, the forest has a secret. Something you can only see in the deepest dark. Something you would have to wait very quietly, very patiently, very still to witness. A glow, a soft, pale, greenish glow, coming from the forest floor. It is not a fire. It does not flicker. It does not warm the air around it. It is cool, steady, continuous. The color of pale moonlight filtered through leaves, or the color of the sea in the very deepest dark, where bioluminescent creatures drift. This is foxfire, the light of bioluminescent fungi, one of the quietest and most extraordinary things the natural world produces. The phenomenon has been known since ancient times. Aristotle, the ancient Greek philosopher, wrote about it over 2,000 years ago. He described a cool light emanating from decaying wood. A light, he noted, that produced produced no heat. That was not fire, and yet glowed like it. Ancient mariners sometimes reported seeing the holds of their wooden ships glow faintly in the night, the wood of the ship being slowly colonized by fungi. The fungi producing their quiet, steady light, lighting the dark interior of the hull with a pale, living green. The source of that foxfire remained mysterious for a long time. It was known to come from decaying wood. It was known to be connected somehow to the growth of fungi on that wood. But exactly how a living organism could produce light without fire was not understood until the science of biochemistry was developed, and what that science revealed is beautiful in its precision. The glow of bioluminescent fungi is produced by a chemical reaction. A reaction involving two main players: a molecule called luciferin and an enzyme called luciferase. The name luciferin comes from the Latin Lucifer, meaning light bearer, the light-bearing molecule. It is the same family of molecules that gives fireflies their summer glow, that lights up the bodies of deep-sea jellyfish in the crushing dark of the ocean, that makes certain bacteria twinkle in the wake of a ship at night. Luciferin, a molecule that, when it meets oxygen and the right enzyme, releases energy as light. In fungi, the reaction works like this. The fungus produces its own luciferin, a specific version unique to fungi. When this luciferin is oxidized, when it meets oxygen with the help of the luciferase enzyme, it releases a photon, a single particle of light, one tiny, fleeting flash of green. But the reaction does not happen just once. It happens continuously, billions of times per second, in billions of fungal cells throughout the mycelium or the mushroom. And the result is a continuous, steady, cool green glow, not flickering, not pulsing, simply there, quiet and constant and pale. All bioluminescent fungi glow in the same narrow range of color, a greenish light at a wavelength of approximately 520 to 530 nanometers. This is not an accident. This wavelength corresponds almost precisely to the peak sensitivity of the night-adapted insect eye, the eyes of moths, of flies, of beetles drawn to the forest floor in darkness. The fungi glow at exactly the color these creatures can see best. In the dark. Why do the fungi glow? This question has fascinated scientists and naturalists for centuries. And the honest answer is that we do not yet fully know. Several explanations have been proposed, and they may all be partially correct. The most widely held idea is that the glow attracts insects. Insects drawn to the glowing mushroom or mycelium, crawling across it, picking up spores on their legs and bodies, and carrying those spores to new locations in the forest. The light on this account is an advertisement, a soft green beacon in the dark, saying, "Come here, find me, carry me forward." For some species, this explanation fits well. The bioluminescent gills of the jack-o'-lantern mushroom, that vivid orange mushroom that glows an eerie green in the dark, are visited by insects in the night. The insects move through the gills, picking up spores, moving them elsewhere. The light serving its apparent purpose. But for other species, the explanation is less clear. Some fungi glow only in their mycelium, deep within decaying wood, where no insect is likely to penetrate, where no light could escape to signal anything outside. For these fungi, the glow may be something else entirely. Perhaps a byproduct of metabolic chemistry, perhaps something that had a purpose long ago and has simply persisted. The forest keeps its secrets. There are approximately 120 known species of bioluminescent fungi. Scientists expect to find more. Bioluminescence is scattered through the fungal family tree, appearing in distantly related groups, suggesting that the ability to produce light has evolved independently multiple times, or perhaps that all bioluminescent fungi share a common ancestor who first developed the trick, and most lineages lost it, while these few scattered survivors kept the light. Some of the most spectacular bioluminescent fungi come from the tropical forests of Brazil. The Ribeira Valley in southern Brazil is home to one of the highest concentrations of glowing fungi in the world. On a warm, damp night after rain, when the humidity is high and the fungi are at their most active, walking through this forest is extraordinary. The fallen logs glow. The leaf litter glows. Small mushrooms the color of cream push up through the dark soil and emit their steady, cool light. The floor of the forest is alive with a pale, shifting luminescence, like starlight caught and held just above the ground. In North America, the phenomenon is known to the bitter oyster mushroom. Panellus stypticus is one of the brightest glowing fungi in the world. It grows in overlapping clusters on fallen logs and stumps. Its small, curved caps, barely wider than a thumb, glow with a brightness that can be seen from several meters away in complete darkness. But only certain strains of Panellus stypticus glow this brightly. The eastern North American strains are luminescent. The European strains largely are not. The same species, the same form, the same delicate fan-shaped cap, but one lineage carrying the light, and one quietly without. The honey fungus, Armillaria, the same genus as that ancient 8,000-year-old organism we spoke of in our very first section, is bioluminescent. Its mycelium glows as it colonizes wood. The rhizomorphs, those dark, shoestring-like strands of compressed mycelium that the honey fungus sends through the soil, glow faintly as they grow. Imagine walking in a forest at night and lifting a piece of bark from a fallen log and seeing beneath it a faint, steady greenish glow. The mycelium of Armillaria quietly working, digesting, decomposing, and producing, as it does so, this cool and ancient light. Foxfire was known to miners in centuries past. Some miners working in deep, dark shafts reported that old timber supports, colonized by fungi, would glow faintly in the dark, providing just enough light to navigate by. A living lantern built not from fire or oil, but from the patient chemistry of fungi on wood. There are stories, too, of foxfire being used by soldiers and travelers, carrying pieces of glowing wood as a light source that needed no flame, that could not start a fire in a dry forest, that glowed steadily and softly through the night. Light provided free and quiet by the forest floor. The forest is dark tonight, and somewhere out there, in the damp wood and the fallen logs, something is glowing, softly, continuously, without heat, without flame. Only the quiet chemistry of living cells, releasing their ancient, pale light into the dark, luciferin meeting oxygen. A photon released, a tiny flash of green, multiplied by billions of cells, into something you could see if you were there, if you were quiet enough, if you waited in the dark long enough. Let that image be the last thing you hold tonight. The glowing forest floor, cool and green and still. The fungi quietly shining. You are warm and safe and completely at rest. Let the light carry you gently forward into the soft, glowing dark. You are doing so beautifully. If sleep has already found you, wonderful. If you are still here, drifting gently on the edge, that is wonderful too. There is no wrong way to rest tonight. Only the slow deepening of quiet, only the forest holding you. Only this voice moving softly through the dark. Tonight, we are going to talk about one of the most extraordinary partnerships in the living world, a partnership so old and so complete that the two partners have become, in a sense, one. Something new, something that could never exist if either partner stood alone. We are going to talk about lichens. Look at an old stone wall or the bark of a very old tree or the surface of a rock at the edge of a mountain stream, and you will almost certainly see them. Patches of gray and green and pale gold and orange, pressed flat against the surface, as thin as paint or raised in small leafy lobes, or standing in tiny, intricate cups. These are lichens, and they are not what they appear to be. A lichen looks like a plant. It grows slowly from a fixed surface. It is often green or gray-green. It is quiet and still and easy to overlook. But a lichen is not a plant. A lichen is not even a single organism. A lichen is a partnership, a collaboration between two very different kinds of living things, most commonly a fungus and an alga. Two organisms living so closely together, so completely intertwined, that they function as one. The fungus provides the body, the structure, the architecture, the tight weave of hyphae that holds everything together and retains moisture and clings to rock and bark. The alga lives within that body, nestled inside the fungal threads, bathed in light filtered through the fungal tissue and the alga photosynthesis. It converts light and carbon dioxide into sugar, and it shares that sugar with the fungus. The fungus gives the alga shelter, protection from drying out, from ultraviolet radiation, from the physical stresses of wind and rain. And the alga gives the fungus food, carbon, the sweet energy of sunlight captured and shared. Two organisms, each providing something the other lacks, each made more by the partnership than either could be alone. The relationship between lichen fungi and their algal partners is believed to have first formed approximately 480 million years ago. This was around the time the first land plants were beginning to emerge from the water. A period of great biological experimentation, of new strategies being tried, new partnerships being formed, and the lichen partnership was one of the most successful experiments in the history of life. Lichens spread across the land. They colonized bare rock. They climbed to the tops of mountains. They spread across the Arctic tundra. They filled the spaces between plants, the niches where nothing else could grow. There are an estimated 20,000 species of lichen in the world. Some scientists think the true number may be higher. Many remain undescribed, living in remote or extreme places that have not yet been fully surveyed. Lichens come in three main body forms. Crustose lichens, the flat ones, pressed tightly against a surface like a coat of paint. You cannot peel them away without destroying them. They are part of the surface they inhabit. Foliose lichens, leafy, loosely attached with lobes that lift slightly from the surface, like small gray-green leaves pressed gently against bark or rock, and fruticose lichens, the three-dimensional ones, standing upright or hanging in pale festoons from branches, like tiny forests, like gray-green chandeliers. Lichens are pioneer organisms. They are often the very first living things to colonize bare rock. When a glacier retreats, for example, leaving behind raw, bare stone that has never known life, the lichens arrive first. They settle on the rock. They secrete weak acids that begin to break the rock surface down. They accumulate organic matter as they grow and die. Slowly, over decades and centuries, they transform bare rock into something that can support other life, the very beginnings of soil, the very first tiny layer of organic matter, from which eventually mosses will grow, and from the mosses, grasses, and from the grasses, shrubs, and from the shrubs, in time, the forest itself. The lichens do not remain. When the forest canopy closes above them, they retreat, returning to the rocks and tree trunks, to the bare, light-filled surfaces where they are most at home. But they laid the groundwork. They prepared the way, quietly as always. The growth of lichens is among the slowest of any organism on Earth. In harsh environments, on high mountain rocks, or in the Arctic tundra, a lichen may grow less than 1 millimeter per year. 1 millimeter, about the width of a pencil mark, in a full year of growing. And yet, because lichens are so slow and so patient and so extraordinarily long-lived, some individuals reach extraordinary ages. A lichen on a rock in the Arctic has been estimated to be over 8,000 years old. 8,000 years. The same age as that ancient honey fungus in Oregon, growing at 1 millimeter per year. For 8,000 years, the mathematics of that patience are almost impossible to hold. Because lichens grow so slowly and so steadily, they can be used to date surfaces. A technique called lichenometry, by measuring the diameter of a lichen on a rock and knowing the average growth rate of that species in that climate, Scientists can estimate when the rock was first exposed, when a boulder was deposited by a glacier, when a wall was built, when a gravestone was placed. The lichen, in its patient circles of pale gray and gold, is keeping a record, a slow, quiet calendar, written in millimeters per year. Lichens are extraordinarily sensitive to air quality because they gather almost all of their water and minerals directly from the air and rain, absorbing them through their entire surface. They have no way to filter out pollutants. Whatever is in the air enters the lichen. Sulfur dioxide, heavy metals, nitrogen compounds. In areas with heavily polluted air, lichens disappear. They cannot survive, and so their presence or absence tells us something important about the quality of the air we breathe. A rock face covered in diverse, healthy lichens is a rock face breathing clean air. A bare rock in a place where lichens once grew may be telling a quieter, sadder story. Some lichens are remarkable for their resilience in conditions beyond anything on Earth. The European Space Agency conducted experiments in which lichen were exposed to the conditions of outer space. Placed outside a spacecraft, exposed to the vacuum, to cosmic radiation, to temperature extremes that would destroy most living things. And when they were brought back inside and given moisture and light, the lichens survived. They began to photosynthesize again within minutes. Organisms older than recorded history, surviving the conditions of space and waking up as if from a nap, as if nothing unusual had happened. There is something in the lichen partnership that is deeply worth sitting with tonight. Two organisms that could not thrive alone, coming together, each giving something, each receiving something, becoming together something neither could be separately, something that can live on bare rock, that can endure the cold of space, that can grow for 8,000 years on a single stone, that can be the first living thing to arrive on a lifeless surface, and the patient, quiet beginning of everything that follows. The lichens on the bark of the forest trees are growing tonight. So slowly, You could not measure it without instruments. A millimeter of growth spread across months. A year of patience written in the tiniest expansion of a gray-green circle, but there, alive, ancient, quietly transforming the surface beneath them. One slow, invisible increment at a time. Let yourself be slow tonight. Let yourself grow at the pace of a lichen, not rushing, not trying, simply being present on the surface of your rest, taking in what the darkness offers, the cool air, the quiet, the deep, unhurried patience of a very old night. You have been growing for a long time, slowly, quietly, in partnership with the world around you. That is enough. That has always been enough. Rest now. The lichens are growing. Still here, still breathing. The night is deep now. Whatever thoughts were troubling you earlier have grown distant, quiet. They are still there, perhaps, but far away, like sounds heard from another room, muffled, soft, not requiring anything from you. Let them be. Tonight, we are going to travel back, much further back than we have gone before. Back through time, through centuries, through millennia, through geological ages with names that sound like music. The Devonian, the Silurian, the Precambrian. Back to a world almost unimaginably different from the one we know. And yet, a world where the fungi were already at work, already quietly, patiently doing what they have always done. The question of how old the fungi are is one that scientists continue to refine. Each decade, new fossils are found. New chemical analyses are performed. New molecular clocks are built. And each decade, the fungi grow older, older than we thought, older than the tools we had last year could measure. For a long time, the oldest confirmed fungal fossils came from rocks approximately 410 million years old. These are the fossils of the Rhynie-Chert, a remarkable deposit of silicified peat in Aberdeenshire, Scotland, where ancient organisms were preserved in such extraordinary detail that scientists can see under a microscope the individual cells of fungi from 400 million years ago. frozen in stone, preserved in the very act of growing, of connecting to the roots of early plants. These fossils show that by 410 million years ago, fungi and plants were already in partnership, already living together in the intimate mycorrhizal relationship we have explored in earlier sections. The relationship was not new even then. It was already established, already ancient, but the fungi were here long before even the Rhynie chert. In 2017, a team of researchers announced the discovery of fossilized fungal-like structures in rocks from beneath the sea floor, rocks dated to approximately 2.4 billion years ago. 2.4 billion years. If that discovery holds up to further analysis, the fungi have been on this planet for nearly half its entire existence. More recent research, using sophisticated molecular dating techniques, comparing the genes of living fungi and tracing back the genetic changes to estimate when lineages diverged, has suggested a common ancestor of all living fungi existing between 900 million and 1.4 billion years ago. And those first fungi were not, it seems, creatures of the land. They were creatures of the water, living in ancient seas and lakes, threading through sediment in the dark, absorbing organic matter, doing in the ancient ocean what their descendants would later do on land in the Canadian Arctic, in rocks dated to approximately 1 billion years ago. Scientists discovered microscopic fossils of extraordinary significance. Tiny, thread-like structures, multicellular, with walls made of chitin, that tough, flexible compound that defines the fungal cell wall. These fossils, named Orisphera geraldi, are among the oldest confirmed fungal fossils ever found. 1 billion years old, preserved in rock in the frozen north, unchanged, waiting to be found for 1 billion years. And in a cave in southern China, scientists found something even more startling. Microfossils in rock dated to approximately 635 million years ago, the oldest known evidence of fungi living on land. Not in the sea, on land, waiting already for the plants that would not arrive for another 100 million years. The fungi were there first, preparing, working the bare rock, building the first thin films of organic matter, laying in their patient and invisible way the foundation of the world. Now let us talk about a creature called prototaxites. It is one of the great mysteries of the fossil record, and even now, scientists continue to debate exactly what it was. Prototaxites. The name sounds ancient, and the creature was. Its fossils span a period from approximately 420 to 370 million years ago, the late Silurian to the late Devonian, a time when life was just beginning to establish itself on land, when the first simple plants were creeping across the bare continents, when the air was thin and strange, when the land was mostly rock and mud. In this stark world, prototaxites stood, not crept, not spread flat across rock, but stood upright like a tree. Its fossils are columns, trunk-like structures up to 1 meter wide and up to 8 meters tall. 8 meters, the height of a 2-story building, standing on a land where no plant had yet reached even a fraction of that height. In its time, prototaxites was by far the largest living thing on land, the tallest, the most visible, a column of living tissue rising from the bare earth, towering over everything. For over 165 years since its discovery, scientists have argued about what prototaxites was. Early researchers thought it was a tree, but its internal structure was wrong for a tree. It had no wood grain, no vascular tissue, no rings. Instead, it was made of interwoven tubes, microscopic, branching tubules, woven together into a solid mass. The internal structure of a fungus, scaled up to the size of a tree. The leading hypothesis for many decades was that prototaxites was a giant fungus, perhaps something like a massive bracket fungus, or a vast, compacted fruiting body, standing 8 meters tall in the Devonian landscape. A fungal tower. The debate has not fully resolved. New research published very recently has cast fresh doubt on the fungal hypothesis. Chemical analyses of prototaxites fossils suggest the molecular fingerprint does not quite match that of known fungi. Some scientists now believe it may represent an entirely extinct lineage of life, something that has no living descendants, something we will never fully know. A mystery written in stone, a column of tubes rising 8 meters from a Devonian shore, and gone now completely, leaving only the fossils and the questions. There is something deeply calming about a mystery that cannot be solved, about a question that simply remains open. We live in a world that often demands answers, certainty, resolution. But prototaxites reminds us that the world has always contained things we cannot fully understand, organisms that do not fit our categories, forms of life that came and went before we arrived to name them. The deep past is full of these quiet mysteries, full of organisms that lived their entire existence without witnesses, without names, without any record except the shape pressed into ancient rock. What we do know with growing certainty is that the fungi were present at almost every major transition in the history of life on land, when the first organisms crawled from the sea, when the first plants took root in bare rock, when the first forests grew, when the great forests fell and became the coal beneath our feet. At every transition, the fungi were there, preparing, connecting, sustaining, transforming. The kingdom that has been here longer than the trees, longer than the flowers, longer, perhaps, than anything else that now walks or crawls or grows on the surface of the land. 1 billion years is not a number that fits inside a human mind. Not really. We can say it. We can write it down. But the full weight of it is beyond our ordinary experience. 1 billion years of fungi, threading through rock and soil and water. 1 billion years of enzymes releasing, of hyphae branching, of spores drifting on whatever currents existed in the ancient world. 1 billion years of quiet, patient, invisible work. And tonight, the same work continues. the soil beneath the forest, in the fallen log softening in the dark, in the white threads still growing, still reaching, still connecting as they have done for longer than the mind can hold. Let that continuity settle over you now. You are resting in a world shaped by 1 billion years of fungal patience. Everything around you, the soil, the trees, the air you breathe, shaped in part by these ancient, quiet, invisible kingdoms. Rest now. Let the deep time hold you. You are in very old hands. Let the night deepen around you now. Let your breathing become even slower, even softer, like the breathing of someone already far into sleep. There is no effort required, only the gentle rising and falling of your chest, only the quiet of this room, only this voice moving softly through the dark. Tonight, we are going somewhere very small. We are going to kneel at the edge of the forest floor. We are going to press our face close to the fallen leaves and look, really look, at what lives there in the world beneath our feet, the world that most of us walk across every day without ever stopping to see. The forest floor has layers, like a very slow, very patient book, each layer telling a different part of the story. The topmost layer is the one you can see. The freshly fallen leaves of this autumn, bright still with some of their color, yellow and brown and deep orange, still holding their shape, still recognizable as the leaf of an oak, or a beech, or a maple. Scientists call this the L horizon. L for litter, the raw material, the beginning of everything. Below the L horizon is the F horizon, the fermenting layer. The leaves here are older. They arrived last year or the year before. They have lost their color. They have lost their crispness. They are soft now, dark brown and beginning to fragment. You can still see, if you look carefully, that this was once a leaf. You can still make out the outline of the midrib, the trace of a vein. But the leaf is becoming something else, something less defined, something in transition. Fungi thread through it. Bacteria colonize every surface. Tiny creatures move through the spaces between fragments. The transformation is underway. Below the F horizon is the H horizon, the humus layer. Here, the original material is unrecognizable. No leaf can be distinguished, no twig identified. Only dark, soft, rich, almost black material. The accumulated work of years, of decades, of centuries in some old forests. This is humus, the most fertile substance in the world, the foundation of all soil health, built grain by grain, layer by layer, by the patient work of fungi and bacteria, and the countless small creatures of the forest floor. Now, let us look closer at what lives in those layers, because the leaf litter is not simply dead material slowly becoming soil. It is a community, a dense, complex, extraordinarily diverse community of living things, all of them going about their lives in the dark, in the damp, in the quiet space between the leaves. 1 gram of healthy forest soil, just 1 gram, the weight of a small paper clip, contains approximately 7,000 species of bacteria, 3,000 species of fungi, and countless representatives of the larger soil fauna. 7,000 species of bacteria in 1 gram of soil. The diversity of life beneath your feet as you walk through a forest is staggering, almost incomprehensible in moist, organic-rich soil. There may be as many as 100,000 springtails in a single cubic meter. 100,000 in a volume of soil roughly the size of a large box. This happens in a fraction of a millisecond. One of the fastest movements in the animal kingdom, in a creature smaller than a grain of rice. Springtails feed on fungal hyphae, on bacteria, on decaying organic matter moving through the spaces between soil particles, between the compressed layers of partially decomposed leaf. There are many thousands of species of soil mites. Some feed on fungi, some on bacteria, some are predators, hunting the springtails and the nematodes. Some feed on the eggs of other mites. A whole world of tiny predation and tiny escape, happening ceaselessly, in total silence, in the dark. The nematodes are even smaller, whole ecosystems in the space between the grains of soil. And the earthworms, those larger, more familiar inhabitants of the soil, moving slowly through the deeper layers, eating organic matter as they go, drawing it down from the surface into the soil, mixing the layers, combining fresh organic matter with mineral soil, and leaving behind in their casts a material extraordinarily rich in available nutrients, a concentrated gift to the roots of plants, the fungi, the bacteria, the entire community of the forest floor. The fungi, of course, are at the heart of all of it, threading through every layer, connecting every level, from the freshly fallen leaf above to the mineral soil below. The fungal hyphae are the infrastructure of the leaf litter, the roads and bridges, the plumbing. Without them, the nutrients in the fallen leaves could not reach the roots of the trees above, could not be cycled back into the living forest. The fungi are the reason the forest floor works as it does. The reason it is not simply a deepening pile of dead material, but a living, breathing, cycling system. There is a detail about leaf litter diversity that scientists have found beautiful in its implications. When a forest floor contains a mix of leaf litter from many different tree species, oak leaves mixed with beech leaves, maple leaves mixed with birch leaves, decomposition happens faster. The diverse mix of chemistry, of textures, of nutritional profiles creates more opportunities for more species of fungi and bacteria, more niches, the diversity rippling from the canopy all the way down through the litter and into the soil. Everything connected, everything influencing everything else. On a warm autumn night after rain, the forest floor is at its most alive. The moisture activates the fungi. New hyphae extend through the wet leaves. The bacteria multiply. The springtails move more actively through the damp soil. The earthy smell, that deep, rich, green smell of the wet forest floor, the dark soil, the rich, quiet, ancient world just beneath the surface. Millions of creatures you will never see. Thousands of species in every handful of earth. All of them working. All of them living. All of them part of the same vast, breathing connected whole. You are resting above all of that tonight, supported by it, held by the long, patient work of the forest floor. Let yourself sink into that support. Let the forest hold you. The leaf litter is soft. The soil is rich and deep. Everything is working. Everything is well. Imagine a forest in autumn. The light is low and gold. The leaves have been falling for weeks. The soil is cool and slightly damp. And somewhere at the base of an old oak tree, perhaps 30 centimeters below the surface, nestled among the roots, something is ready. It has been growing slowly since spring, building itself cell by cell in the dark. And now, at last, it is ripe. It cannot move. It cannot open to release its spores on the breeze. It is trapped underground with no way out. And so it does the only thing it can. It calls, not with sound, with scent. The truffle is one of the most astonishing organisms in the fungal kingdom. Not because it is rare, though some species are. Not because it is valuable, though it certainly is, but because of the particular, precise, extraordinary solution it has found to the problem of being a fungus that cannot release its spores into the air. It grows underground, completely. Its entire fruiting body, the structure we know as the truffle, forms beneath the soil surface, tucked among the roots of its tree partner, hidden from view, hidden from wind, and it relies on the animals of the forest to find it, to dig it up, to eat it, and carry its spores to new places. Truffles are mycorrhizal fungi. They belong most famously to the genus tuber, and they form ectomycorrhizal partnerships with specific trees, most commonly oaks, hazelnuts, beeches, pines. The partnership is the same fundamental relationship we explored when we talked about the wood wide web. The fungus provides the tree with water and minerals. The tree provides the fungus with sugars. A mutual exchange, a slow, patient bargain struck in the dark soil. And the truffle is the fruiting body, the brief, dense, reproductive structure that forms when conditions are exactly right. The interior of a truffle is a maze. If you were to cut one open, you would see a pattern of pale veins running through darker flesh, like the marble in stone, or the network of streams seen from above. These veins are the sterile tissue, the structural scaffolding between the veins, packed densely, are the spore-bearing cells, the asci, each ascus containing multiple spores, protected within the tough, enclosed body of the truffle, safe from the weather above, safe from drying out, waiting in the dark for an animal to find them. The scent is the signal. When the spores inside a truffle are mature and ready for dispersal, the truffle begins to produce volatile compounds, dozens of them, sometimes more than 100 distinct chemical molecules, each contributing to that complex, layered, extraordinary aroma. The scent diffuses slowly through the soil, filtering up through the leaf litter, rising into the air above the forest floor. Faint, subtle, almost imperceptible to human senses, but to the nose of a pig, or a trained dog, or a red squirrel, or a flying squirrel, or any of the dozens of forest animals that have evolved to seek out and eat truffles. The scent is unmistakable, a clear, rising signal. Here, underneath, come and find me. Female pigs were among the first animals employed by humans in the search for truffles. They were drawn to truffles instinctively, not trained, simply drawn, because certain compounds in the truffle scent resemble androstenol, a pheromone produced by male pigs. The female pig, following her nose through the forest, was in some sense being fooled, searching for something that smelled, to her, of a potential mate, finding instead a truffle. The practice of using pigs to find truffles is documented as far back as the Roman Empire. And by the 15th century, the truffle hog was a well-established part of European food culture. But pigs had a significant drawback. Once they found the truffle, they wanted to eat it immediately. And a female pig is a large, determined creature, difficult to redirect when she has found something she wants. The truffle hunter had to be quick, and even then, pigs would sometimes consume the truffle before it could be retrieved. In Italy, the use of pigs for truffle hunting was eventually prohibited in 1985 to protect the mycelium from the damage caused by enthusiastic pig digging. Dogs replaced the pig, not because dogs have a better nose. They do not. A pig can detect a truffle buried 3 meters deep. A dog typically detects truffles buried to about 1 meter. But dogs are trainable. They can be taught to indicate the location of a truffle without consuming it, to sit or to paw gently, or to look back at their handler, waiting for their reward. A small treat, a moment of praise, the scent of the truffle exchanged in the dog's calculation for something more immediately satisfying. Training begins when the dog is just a few months old. The relationship between a truffle hunter and a trained dog is a genuine partnership. A quiet, slow collaboration built over months of patient work. The dog's extraordinary nose, the hunter's knowledge of the forest, of which trees, of which soil, of which conditions. Together, they find what neither could find alone. In the wild forest, of course, no human is needed. The animals find the truffles without any guidance. Squirrels, deer, wild boar, insects, even some birds, all of them drawn in different ways by the scent, digging, eating, carrying the spores in their digestive tracts to distant parts of the forest, depositing them in their droppings. Each deposit a potential new beginning, a spore left in a new place, near new roots, with new opportunities to germinate, to grow, to form new partnerships. The truffle, unable to move, having arranged for the world to carry it forward. There are hundreds of truffle species. Most are not the celebrated Perigord black truffle or the white truffle of Alba. Most are modest, underground, unknown to all, but the most dedicated mycologists in the Pacific Northwest of North America. A particularly diverse community of truffle species lives beneath the conifer forests. Douglas fir trees their partner with an extraordinary variety of ectomycorrhizal fungi. One researcher estimated that a single Douglas fir might associate with as many as 2,000 different fungal species. Many of these form underground truffle-like bodies, small, unassuming, smelling of nothing to us, but deeply significant to the small mammals of the forest. The flying squirrels, the voles, the deer mice, who rely on these underground fungi as a significant part of their diet, especially in winter, when other food is scarce and the truffles stored underground remain available, hidden beneath the snow. There is a beautiful circularity to the truffle story. The tree feeds the truffle. The truffle feeds the animal. The animal spreads the truffle's spores. The spores germinate and form new mycorrhizal partnerships. The new fungus feeds a new tree. The new tree feeds the new fungus. On and on, round and round. The forest sustaining itself. It has been building itself since spring, patient and unhurried. It does not know it will be found. It does not plan. It simply grows and scents and waits. The forest knows what to do. The animals will come. The spores will travel. The partnerships will form. The forest will continue. Let that quiet confidence carry you into deeper sleep now. Everything is unfolding as it should. The truffle is waiting in the dark, and somewhere in the forest, a nose is lifting, catching something on the cool night air, something subtle and rich and extraordinary, following it toward the soil, toward the hidden treasure beneath the roots. Let your thoughts drift like that scent, gently upward, and then dissolving into the cool forest dark. Still here, still breathing, that slow, steady rhythm. Let it deepen a little more with each breath. Let the night settle a little more heavily over you. Tonight, we are going even deeper than we have gone before. Not into history, not into distant forests, but into the very ground itself, into the structure of soil, into the hidden architecture that fungi have been building for hundreds of millions of years. The architecture that holds the world together. Soil is not simply ground up rock. It is not just dust and sand and mineral particles. Soil is a living structure, a built thing, assembled over vast stretches of time by the combined work of countless organisms, and among all of those organisms, the fungi have a role that is unique, a role that cannot be replicated by anything else, a role that determines to a significant degree whether soil holds together, whether it holds water, whether it supports life, whether it is soil at all, or only sand. The key to understanding what fungi do to soil lies in something called aggregation. Soil aggregates are small clumps of soil particles, not individual grains of sand or silt or clay, but groups of particles bound together, held in loose, stable clusters. These clusters, these aggregates, are the basic building blocks of healthy soil structure. When a soil has good aggregation, it has spaces between the aggregates, pores, tiny channels and cavities where air can move, where water can percolate, where roots can push through, where the microscopic organisms of the soil food web can live and move. When a soil lacks aggregation, those spaces collapse. The particles pack tightly together. Water runs off rather than soaking in. Roots struggle to penetrate. The soil, in a sense, suffocates. Fungal hyphae help create and maintain soil aggregates in two ways. The first is mechanical. As hyphae grow through soil, they physically thread through and around soil particles, wrapping around them, binding them together, holding the aggregate in place, the way a net holds fish, or the way roots hold a riverbank. A single hypha is extraordinarily fine, but mycelium is a dense, branching network, and through a handful of healthy soil, millions of meters of hyphae may be woven, holding the structure together, preventing it from collapsing into a formless mass. The second way fungi build soil aggregates is chemical, and this involves one of the most remarkable substances in the soil. A substance that, for most of the 20th century, was completely unknown to science. It was only discovered in 1996. A researcher named Sarah Wright, working at the USDA Agricultural Research Service, found something in soil samples she couldn't account for. A protein, persistent, widespread, present in soils all over the world, and it was coming from the fungi. She named it glomalin. Glomalin is a glycoprotein, a protein with a carbohydrate chain attached, produced by arbuscular mycorrhizal fungi and deposited into the soil as those fungi grow and eventually die. It is extraordinarily stable. Its complex molecular structure resists decomposition. It can persist in soil for decades. Some estimates suggest up to 100 years. And in that time, it does something remarkable. It acts as a glue. Glomalin coats fungal hyphae. It coats soil particles. It binds them together, sticky, water-resistant, durable, like a natural cement, spread through the soil by the patient work of mycorrhizal fungi. The soil particles, coated in glomalin, clump together into stable aggregates. Aggregates that hold their shape even when wet, even when rain falls hard, even when the soil is disturbed. The glomalin keeps them together. The structure persists. Scientists estimate that glomalin may comprise as much as 2% of total soil weight. That does not sound like much, but 2% of the world's soil is an enormous quantity, and its importance far exceeds its proportion. Glomalin contains carbon, significant amounts of carbon locked within its stable, resistant structure, held in the soil for decades, protected from decomposition. The carbon that plants absorb from the air, that flows down through roots, that is passed into mycorrhizal fungi, that is incorporated into glomalin and deposited into the soil, where it stays, for years, for decades, building the slow, deep carbon store of the forest soil. Healthy fungal networks also regulate the water in soil. This is a subtle and extraordinary thing. Mycelium holds moisture. The fine threads of hyphae, with their large surface area, absorb water and hold it within the network. In dry conditions, mycelium can act like a sponge, retaining moisture in the soil, slowing evaporation, keeping the water available for plant roots and soil organisms. But mycelium also produces hydrophobic compounds, water-repelling substances that coat the outer surfaces of hyphae. And these compounds change the way water moves through the soil. They slow infiltration. They help the soil hold on to moisture at depth, preventing it from percolating too quickly through sandy soils into the groundwater below. The mycelium, in this way, acts as a kind of water manager, holding moisture where it is needed, releasing it slowly, regulating the rhythm of water through the forest floor. Think for a moment about what all of this means for a forest. The trees reach down with their roots. The mycorrhizal fungi reach out from those roots, threading through the soil, building aggregates, depositing glomalin, holding moisture, creating the pore structure that allows air and water to move, providing the physical infrastructure without which the soil would compact, without which the rain would run off rather than soak in, without which the roots would struggle, without which the trees themselves could not grow as tall or live as long. The fungi are not simply feeding the trees, they are building the very ground the trees stand on. In an old-growth forest, the soil can be extraordinarily deep and rich. Centuries of fungal work, layer upon layer of glomalin, aggregate upon aggregate, a structure so stable and so complex that it can hold water in a drought, can absorb the heaviest rain without flooding, can sustain the roots of enormous trees through the hottest summers. This is not an accident. It is the result of hundreds of years of patient, continuous fungal activity, building the soil, grain by grain, season by season, in the quiet dark beneath forest floor. When forests are cleared, when the trees are removed and the soil is exposed and the mycorrhizal networks are broken, something important is lost. Not just the trees themselves, not just the shade and the habitat, but the soil structure without the trees providing sugars to the fungi, and without the fungi building and maintaining the aggregates. The soil begins to change. Aggregates break down. Glomalin levels fall. The soil becomes less stable, less able to hold water, more vulnerable to erosion. The rain falls, and instead of soaking in, it carries the soil away. The surface of a forest, built over centuries, can be lost in a few seasons of rain without the fungal networks holding it together. This is why the fungi beneath a forest are not merely interesting. They are essential. The invisible infrastructure of a living world. The glue that holds the soil together. The water that holds back the drought. The structure that makes room for roots and air. And the countless small creatures of the earth. Tonight, as you rest, the soil beneath forests across the world is being built. Grain by grain, thread by thread, molecule of glomalin deposited quietly by a hyphal wall, binding one soil particle to the next, building the aggregate, building the structure, building the ground, slowly, without ceasing, in the patient, unhurried dark. Let that steadiness move through you now. That slow, quiet building, that grain by grain patience. You are held by something very old and very strong. The ground beneath the forest is firm. The fungi are working. Everything holds. Still here, breathing. The night is very deep now. You have been resting for a long time, and the forest has been resting with you. The trees standing still in the dark, their leaves barely moving, their roots deep in the cool soil. And between those roots, in the dark, patient network of mycelium beneath the forest floor, something vast and quiet is happening. Something that touches every living thing on earth. something that connects the breath of the forest to the air we breathe, to the sky above, to the slow, invisible chemistry of the atmosphere. Tonight we talk about carbon and the quiet, extraordinary role that fungi play in holding it. Carbon is the element of life. Every living thing is built from it. Every cell, every molecule of protein and fat and sugar, every strand of DNA, every thread of mycelium, every leaf on every tree, everything alive is, at its most fundamental level, a structure of carbon atoms arranged in particular ways. And carbon does not stay in one place. It moves constantly from the air to the plant, from the plant to the soil, from the soil to the atmosphere, from the atmosphere back to the plant. A cycle that has been turning for hundreds of millions of years, the carbon cycle. And fungi are among its most important participants. Let us follow one carbon atom. It begins as carbon dioxide, a molecule floating in the atmosphere above the forest. A tree's leaf absorbs it, captures it from the air in the process of photosynthesis, combines it with water, uses the energy of sunlight to turn it into sugar, a simple glucose molecule. Carbon, now in a new form, held within a living cell. The tree uses some of that sugar immediately for energy, for building new tissue, but a significant portion, somewhere between 5 and 30% of all the carbon a tree fixes from the air, is sent downward, down through the trunk, down through the roots, into the mycorrhizal fungi, into that vast, patient network of mycelium threaded through the soil. This is the carbon payment, the tree's contribution to the partnership, the cost of the nutrients and water the fungi provide. And it is substantial. When scientists measured how much carbon the world's plants send to mycorrhizal fungi each year, the number was extraordinary, an estimated 13 billion tons of carbon dioxide equivalent. Every year, 13 billion tons. This is equivalent to approximately 36% of current annual global fossil fuel emissions. More carbon passing from the world's trees and plants into the soil fungi beneath them each year. Then China emits from all its industry and transport combined. Let that number settle gently, not as a source of alarm, as a source of wonder. The trees and plants of the world are constantly, quietly breathing carbon downward into the soil, into the fungi, vast quantities of it, every year, every season, without ceasing. Now, what happens to that carbon once it enters the fungal network? Some of it is used by the fungi themselves for growth, for building new hyphae, for producing enzymes and proteins and cell walls, for all the metabolic work of a living organism. But some of it enters the soil. And here is where the story becomes extraordinary. When mycorrhizal fungi grow, they shed fragments of hyphae. Fungal cell walls are made of chitin, that tough, resistant material. And chitin is slow to break down, slower than plant tissue, slower than leaves, much slower than simple sugars. The carbon locked within those dead hyphae stays in the soil for months, for years, for decades. A remarkable study of boreal forests in Sweden found something that upended decades of scientific understanding. The conventional wisdom had always been that the main source of carbon storage in forest soils was dead leaves and wood falling from above, the litter layer, the accumulated debris of the forest floor. But when these researchers looked carefully at the Swedish islands they were studying, they found something different. 50 to 70% of the carbon stored in the leaf litter and soil was not coming from above. It was coming from below, from the roots, and from the mycorrhizal fungi associated with those roots. Carbon that had flowed down from the trees into the fungal network and been deposited into the soil as fungal biomass. The fungi were the primary pathway, the main route by which living carbon entered the soil carbon pool, not the falling leaf, not the rotting log, but the quiet, steady, invisible work of the mycorrhizal network, depositing itself into the soil, season by season, thread by thread, and the type of fungi present in a forest matters. A study published recently, examining forest soils across the European continent, found that the composition of the fungal community, which species of fungi were present, and in what proportions, was a strong predictor of how much carbon the forest stored. More predictive, even, than the tree species themselves. More predictive than the climate. Forests with diverse, healthy fungal communities stored more carbon, grew their trees faster, accumulated more biomass. The fungi were not merely following the forest. They were, in some measurable sense, creating it. There is a nuance worth holding here. Not all fungi store carbon equally. Some fungi, particularly the saprotrophic decomposers we spoke of earlier, release carbon from the soil. They break down organic matter, release carbon dioxide. They are, from the carbon perspective, releasers rather than storers, and some mycorrhizal fungi, particularly those in nutrient-poor soils, can also break down soil organic matter to access the nitrogen locked within, releasing some carbon in the process. The balance between storing and releasing, between the fungi that build the soil carbon pool and those that decompose it, is one of the most active areas of research in forest ecology. And the answer is not simple. It is, like so much in the fungal world, a matter of balance, of relationship, of context. What is clear is that when forests are healthy, when their mycorrhizal networks are intact and diverse, the fungi act at scale as one of the most significant carbon stores on the planet. More carbon flowing into the soil through fungal networks each year than through almost any other pathway. The forest breathing inward, pulling carbon from the sky, sending it down through the roots, into the mycelium, into the soil, held there quietly in the dark. This is the breath of the forest, not the visible breath, not the oxygen released from leaves into the summer air, but the other breath, the downward breath, the slow, invisible exhalation of carbon into the soil, the forest breathing in and passing its breath downward into the earth, where the fungi receive it, hold it, build themselves from it, and leave the residue in the soil when they die. Layer by layer, year by year, building the deep, dark, carbon-rich humus of the old forest floor. Tonight, the forests of the world are breathing. Every tree, every mycorrhizal partnership, every thread of mycelium receiving carbon from its tree partner, 13 billion tons of it in the course of a year, flowing silently downward into the dark, into the soil, into the slow, patient storage of the earth. You are breathing too, slowly, quietly, carbon moving through you, in and out, part of the same vast cycle, the same turning wheel that has been turning since long before the first tree stood. Let that connection be the last thought you hold tonight. You are breathing with the forest, carbon moving gently in and out of the world, quietly holding its breath, and you resting within it, safe, still, deep. You are still here, still breathing slowly. The night is very deep now, very still. Let your body be completely heavy. Let your arms and legs sink. Let your shoulders fall. Let even the small muscles of your face soften. There is nothing to hold, nothing to carry. Only the quiet, only the dark, only this gentle voice moving through the forest with you. Tonight, we are going to talk about something that has been quietly saving lives for thousands of years, something that grew on the bark of trees, in the crevices of logs, on the walls of old caves, something that people looked at long ago and somehow knew, somehow understood before science could explain why. That this was medicine, that this was healing. The fungi, quietly, have always been healers. Long before laboratories, long before clinical trials, long before the word antibiotic existed, people around the world were using fungi as medicine. In China, the first formal written record of medicinal mushrooms dates to approximately 2,000 years ago. A text called the Shen Nong Ben Cao Jing, the classic of herbal medicine, written during the Han Dynasty. It describes dozens of medicinal plants and fungi, and among them, one mushroom in particular is given the highest possible distinction. It is listed in the superior class of medicines, the class reserved for substances that, taken over time, promote longevity, calm the mind, and sustain the body. That mushroom was Rhizae, Ganoderma lucidum, the mushroom of immortality. Rhizae grows on the decaying wood of broadleaf trees. It is a bracket fungus. Its surface is hard and lacquered looking, deep reddish brown, almost glossy, like polished wood. It does not look like something you would eat, and in fact, raw Rhizae is extraordinarily bitter, almost unpalatable, but prepared as a tea, simmered slowly in water for hours. The compounds within it dissolve into the liquid, and the resulting brew was drunk for centuries as a tonic, a strengthener, a calmer, a thing that settled the mind and supported the body. In ancient China, Rhizae was so revered that it appeared in art, in architecture, in the robes of emperors, in the carvings on palace furniture. The mushroom of immortality, emblem of health and long life. Modern science has begun to understand why. Rhizae contains compounds called polysaccharides, specifically beta-glucans, complex sugar molecules that interact with the immune system, signaling to immune cells, modulating their activity, encouraging the body's defenses to be alert and responsive. It also contains triterpenes, bitter, complex molecules that may have anti-inflammatory properties that may support liver function, that may help regulate blood pressure. The science is careful, the evidence in some areas still developing, but the compounds are real. Their effects on human cells measurable. The ancient healers of China were not wrong to value this mushroom. They simply did not have the vocabulary to explain why it worked. Chaga is another fungus with a long history as medicine. It grows on birch trees in the cold forests of Russia, Siberia, Scandinavia, and northern North America. It does not look like a mushroom at all. It looks like a piece of burnt charcoal, a dark, rough, irregular mass protruding from the bark of a birch tree. Inside, it is orange-brown, rich with compounds, and it has been used as medicine in Siberia and Russia since at least the 16th century, prepared as a tea, drunk slowly through the long winter, a warmth from the birch tree, a medicine from the cold forest. The compounds within chaga include enormous quantities of antioxidants, some of the highest concentrations of antioxidants measured in any natural substance, and polysaccharides and betulin and betulinic acid, compounds derived from the birch tree that the chaga has been colonizing. Compounds now being studied for their effects on cancer cells, on inflammation, on the regulation of blood sugar. The forest pharmacy, growing quietly on the white bark of a birch in a cold wood, waiting for the healer who knows where to look. Turkey tail, that beautiful fan-shaped fungus you sometimes see growing in overlapping tiers on fallen logs, its surface banded in concentric rings of brown and cream and gray, like the tail feathers of a wild turkey fanned in display. Turkey tail has been used in traditional medicine across Asia for centuries, and it contains a compound called polysaccharide K in Japan. This compound has been approved as an adjunct cancer treatment, used alongside conventional chemotherapy to support the immune system during treatment, to help the body withstand the rigors of the therapy. It is the only mushroom-derived compound to have reached this level of clinical approval. And then there is lion's mane, Hericium erinaceus, one of the most striking mushrooms in the temperate forest. It grows on the wounds of old hardwood trees, producing a mass of long, cascading white spines, like a waterfall frozen in white, or the flowing mane of a pale, gentle animal. It has been used in traditional Chinese medicine for centuries, and modern research has found in it something extraordinary. Compounds called hericenons and erinacines, small molecules capable of crossing the blood-brain barrier and stimulating the production of nerve growth factor, a protein that encourages the growth and maintenance of nerve cells in the brain. Scientists are studying these compounds with considerable interest as possible approaches to neurodegenerative conditions. to the protection of the aging brain. The forest, once again, offering something that no laboratory had previously imagined, but of all the gifts that fungi have given to human medicine, perhaps the most transformative is the one discovered not in the ancient forests of China, not by a traditional healer moving through winter woods, but in a London laboratory. In the early morning, in September of 1928, Alexander Fleming had returned from a summer holiday. He was sorting through old Petri dishes in his laboratory at St. Mary's Hospital, dishes that had been left on the bench while he was away. And on one dish, a dish that had been growing a colony of Staphylococcus bacteria, he noticed something. A mold had grown. It had blown in through an open window, perhaps, or drifted from the floor below, where a colleague was studying molds. And around the mold, the bacteria were dead. A clear zone, a circle of absence in the living colony. The mold had produced something that killed the bacteria, something that bacteria had no defense against. Fleming identified the mold as Penicillium notatum, and he named its active substance penicillin. The development of penicillin into a medicine took another decade. It required the work of Howard Florey and Ernst Chain at Oxford. It required the war, the desperate need for something that could treat infected wounds. And it required a supermarket cantaloupe. In 1943, a laboratory worker named Mary Hunt brought in a cantaloupe she had found in a market. It was infected with a mold that had, she noted, a pretty golden look. This mold turned out to be a strain of penicillium that produced far more penicillin than Fleming's original strain. It became the basis for the industrial production of the antibiotic. The mold from a cantaloupe, changing the course of medicine, saving an estimated 200 million lives in the decades since, all from a spore that drifted in through a window onto a forgotten Petri dish. In the summer of 1928, fungi have given us other medicines too. Statins, the cholesterol-lowering drugs taken by hundreds of millions of people worldwide, were originally derived from a fungus called penicillium, citrinum, cyclosporin, the drug that makes organ transplants possible by preventing immune rejection, comes from a soil fungus called Talipocladium inflatum. The forest floor, quietly offering its chemistry, the quiet, hidden world of fungi, producing compounds that human chemists would never have thought to invent. The forest pharmacy is still largely unexplored. Of the millions of species of fungi estimated to exist on Earth, only a fraction have been studied for their chemical compounds. Only a tiny fraction of those compounds have been investigated for medicinal properties. Scientists working with new analytical tools are finding in every forest soil sample they examine molecules they have never seen before, compounds built by fungi over millions of years of evolution, compounds shaped by the need to compete, to defend, to communicate, to survive. Some of those compounds will have no use to us, but some will. Some already have, and the forest, patient as always, is holding the rest, waiting. Rest now. The forest is full of healers tonight, on the bark of birch trees, on the fallen logs, in the soil, in the dark between the roots. Compounds older than our medicine, older than our science, old as the forest itself. You are resting in a world that heals, that has always healed, slowly, quietly, without asking anything in return. Let yourself be held by that. Still here, still breathing. You have come a long way tonight, through the threads of mycelium, through the wood wide web, through the fairy rings and the glowing dark, through the deep, slow soil, through the ancient history of the fungal kingdom. And now, as the night deepens toward its quietest hour, we arrive somewhere very old, very still, a place that takes the breath away if you let yourself really feel it. An old-growth forest. Old-growth forests are rare in most parts of the world. The forests we see are young, planted in the last century, or regrown after clearing. Trees of 50 years, 100 years, even 200 years. These are not old growth. True old growth forests forests that have never been cleared have been growing without significant human disturbance for centuries, some for millennia. And they are not simply older versions of younger forests. They are something fundamentally different, a different kind of place, a different order of complexity. In an old-growth forest, the trees are enormous, some of the largest living organisms on Earth. Douglas firs in the Pacific Northwest, reaching over 90 meters tall, redwoods taller still. Ancient Alerce trees in the temperate rainforests of southern Chile, estimated to be over 3,000 years old. Beeches in European old-growth reserves, centuries in the making, their bark thick and ridged and covered in mosses and lichens, and the fungal communities around their roots are unlike anything found in younger forests. A recent study from the University of Melbourne examined the soil around ancient Alerce trees in Chilean Patagonia, trees over 2,000 years old. Trees so large their trunks measure several meters across at the base. The researchers sampled the soil around these ancient giants, and what they found was extraordinary. A single large Alerce, one individual tree, supported more than twice the underground fungal diversity found in the soil around smaller, younger trees of the same species. The largest tree they sampled hosted more than 300 fungal species, unique to that one individual species found nowhere else in the forest. Species possibly found nowhere else on Earth. 300 hidden species living quietly in the soil around one ancient tree, some of them likely unknown to science. This finding has profound implications. If a single ancient tree can shelter 300 unique fungal species, and if many of those species are found nowhere else, then when that tree is lost, those fungal communities may be lost with it. Not just one tree, not just 300 years of growth, but 300 unique species, an entire underground ecosystem assembled over millennia. Gone, the researchers put it plainly. Losing a single millennial tree could mean losing an entire hidden ecosystem that took thousands of years to assemble. Old-growth forests are characterized by structural diversity, not just the living trees, but the dead ones too. The standing dead trees, the snags, the fallen logs in every stage of decay, the nurse logs, the root mounds where trees have tipped over, pulling the soil up with them, creating small hills of exposed mineral earth, and hollows of rich, organic debris beside them. This structural diversity is not disorder. It is complexity. And it is precisely this complexity that the fungi require. Different species of fungi need different substrates, different stages of decay, different microhabitats. Some species only colonize the heartwood of ancient standing trees. Some only grow from logs that have been decomposing for at least 50 years. Some require the particular chemistry of a root mound, where mineral soil has been mixed with organic matter, all of them dependent on the full, complex range of conditions that only an old-growth forest provides. One such species is the agarican. Laricifomes officinalis, a bracket fungus that grows only on the trunks of old, large conifer trees. In the Pacific Northwest of North America, it produces enormous fruiting bodies, white and chalky like an ancient hoof growing from the side of a fir or larch. Some agarican brackets grow for decades, accumulating layer by layer, like the rings of a tree, recording the years. It is listed as endangered, found only where the oldest trees remain. Without old-growth forest, agarican cannot exist. It is entirely dependent on the conditions that only centuries can create. In Northern Europe, scientists studying old-growth forest reserves have found a consistent pattern. Forest naturalness, measured by the volume of dead wood, the age of the canopy trees, the continuity of forest cover over time, is the strongest predictor of fungal diversity. Old forests have more species, more rare species, more species found nowhere else. And this diversity cannot be quickly recreated by simply planting trees. A forest of young planted trees, however dense, however carefully managed, uninterrupted forest continuity has built over centuries. The fungal networks in old forests are not simply larger, they are more connected, more complex, more redundant, more capable of maintaining their function under stress. There is a concept in ecology called redundancy. It sounds technical, but it is, in its essence, a beautiful thing. Redundancy in an ecosystem means that multiple species can perform the same function. If one species of fungus that decomposes a particular type of wood is lost, another species can step in. The function continues. The forest does not collapse. But this redundancy, this insurance against loss, only exists when there are enough species, enough diversity. Old growth forests have that diversity. Young forests, regrown after clearing, often do not. The young forest may look like a forest. The trees may be tall. The canopy may be closed. But underground, the network is simpler, less connected, less resilient. The trees more vulnerable to stress and disease and drought because the ancient, complex fungal foundation has not yet been rebuilt. When a seed germinates in an old growth forest, it does not arrive alone. It puts down its first roots into a soil already threaded with mycelium, already connected, already a part of the network. The fungal threads find the new root. The mycorrhizal partnership forms within days, and the seedling immediately gains access to the resources of the entire network. The nutrients, the water, the chemical signals, the support of the mother trees above. In a plantation or a regrown forest, that network is absent or thin. The seedling must find its own way. In the rich complexity of old growth, it is welcomed into a community from its very first day. This is why old growth forests matter so much. Not simply for their size, not simply for their carbon, not simply for the rare species they shelter above ground, but for what lies beneath the ancient, complex, irreplaceable fungal networks built over centuries, connecting everything, supporting everything, sustaining the forest through droughts and storms and the slow changes of climate. networks so old and so complex that we are only beginning to understand what they do and what would be lost without them. Tonight, somewhere in the world, an old-growth forest is standing. A forest that has never been cleared, never been logged, never been reduced to bare ground and replanted from scratch. A forest where the trees are centuries old, where the fungal networks beneath them are centuries old, where the soil has been built and rebuilt, grain by grain, for longer than any living person can remember. Let that forest be the image you carry into your deepest sleep. The tall, still trees, the soft floor of ancient leaves, the pale threads of mycelium in the dark soil. Old beyond imagining, connected beyond counting, quiet beyond words. You are resting in the memory of that forest tonight, held by something ancient, something that has survived everything the world has thrown at it, simply by growing slowly, by staying connected, by holding together. Let that be enough. Let yourself be held. You have come so far tonight, through the ancient kingdom, through the mycelium and the networks, through the fairy rings and the glowing dark, through the decomposers and the healers and the oldest organisms on earth. And now we arrive at the final chapter. Not an ending. Nothing about the fungal world ends neatly, but a turning, a facing forward, a quiet look at what comes next, at the ways this ancient kingdom may help shape the world that is still becoming. Tonight, we talk about fungi and the future. And it is, despite everything, a hopeful story. The world we live in is facing challenges. Soil exhausted by decades of intensive farming. Forests cleared faster than they can regrow. Pollutants accumulating in places that were once clean. Plastic drifting through oceans. Heavy metals leaching through soil into groundwater. The long, slow bill coming due for a century of industrial activity. These are real problems, heavy, complex, not easily solved. But the fungi have been solving versions of these problems for hundreds of millions of years. And scientists are beginning to pay close attention. The use of fungi to clean contaminated environments has a name: mycormediation, from the Greek mykes, meaning fungus, and the Latin remediare, meaning to heal, fungal healing. The idea is simple in principle. The fungi that have evolved to break down the most complex organic molecules on Earth, the ones that can dissolve lignin, that tough structural polymer that almost nothing else can touch, can also break down many of the complex organic pollutants that humans have introduced into the environment. Petroleum products, industrial solvents, pesticides, certain plastics. The white rot fungi are particularly capable. The same oyster mushroom that fans out in pale tears from a fallen log. The same Trametes versicolor, the turkey tail, that we met in our section on healing. These fungi produce enzymes so powerful, so adaptable, that they can attack and break down molecules that no naturally occurring process would easily dismantle. In one experiment, contaminated soil was inoculated with oyster mushroom mycelium. Soil heavily polluted with diesel and petroleum products. After just 8 weeks, the pollution had been reduced dramatically. The mycelium had threaded through the soil. Its enzymes had gone to work, breaking the long hydrocarbon chains, dismantling the petroleum compounds, transforming them into simpler molecules: carbon dioxide, water, and the ordinary organic matter of a functioning soil. And as the mycelium did its work, mushrooms fruited from the contaminated ground. The mushrooms attracted insects. The insects attracted birds, and slowly, around this small patch of healing soil, life returned. Fungi can also accumulate heavy metals. This process is called biosorption. The hyphae of certain fungi bind to metal ions in contaminated soil or water: copper, cadmium, lead, mercury. The metals are drawn onto the surface of the hyphae or absorbed into the fungal tissue, sequestered there, removed from the soil or water where they were causing harm. The fungus can then be harvested, removed from the site, the metals taken with it, leaving behind soil that is cleaner than before. Scientists in Mexico City found something remarkable when they introduced oyster mushroom mycelium to used diapers. Diapers are notoriously difficult to decompose. Their combination of synthetic polymers, absorbent gels, and cellulose resists most natural breakdown processes. But the oyster mushroom went to work. After just 2 months, the mycelium had reduced the mass of the diapers by 85%. 85% compared to just 5% in the control group, where no fungi were present. The same enzymes that unravel the lignin in a fallen oak can, it turns out, be turned to dismantling the synthetic materials we have made. Given enough time, given the right conditions, given the patient, unhurried attention of the fungi, and then there is what mycelium can build. In recent years, a new generation of designers and engineers and material scientists have begun to work with mycelium not as a cleaner of pollution, but as a builder of things, mycelium-based materials, growing in molds, shaped into packaging, into insulation, into furniture, into the soles of shoes. The process begins with agricultural waste, corn husks, hemp fiber, sawdust, materials that would otherwise be discarded. The waste is inoculated with mycelium and then simply left. For a few days, the mycelium grows through the substrate, binding it, weaving through every particle, creating, as it grows, a composite material, dense, strong, lightweight, fully biodegradable. At the end of its useful life, a mycelium-based product can be composted, returned to the soil, decomposed by the same fungi that built it, the carbon in the packaging, released back into the cycle. Nothing wasted, nothing persisting in a landfill for 500 years, just the quiet return, the circle completing itself. A company in New York has used mycelium packaging to replace polystyrene in the protective packaging of electronics. Dell has shipped servers cushioned in mushroom packaging, growing in 5 to 10 days, using no fuel beyond the carbohydrates of the mycelium itself, decomposing in a compost heap within researchers at the University of Maine have developed mycelium coatings for paper, a film just 20 to 25 microns thick, roughly a quarter of the thickness of a human hair. That provides moisture resistance comparable to plastic, but decomposes naturally when the packaging is discarded. The coating forms a faint, slightly fuzzy surface on one side, like the texture of a mushroom cap, soft to the touch, alive in a way with the residue of its making. And in ecological restoration, the fungi are proving to be some of the most important partners available when agricultural land is returned to nature, when the plowing stops and the land is allowed to rest. The mycorrhizal fungi return faster than many scientists expected. Research from the University of Guelph found that arbuscular mycorrhizal fungi can bounce back within 10 years of restoration. 10 years. In ecological terms, extraordinarily fast. The fungal spores persist in the soil through years of farming, waiting, patient, as fungi always are. And when conditions become favorable again, when roots return and organic matter accumulates, and the soil is disturbed less often, the fungi germinate, colonize the new roots, rebuild the network, begin again. The forest, given the chance, remembers what it was. The fungi hold that memory in their spores, in their chemistry, in the resilient, ancient mechanisms of germination and growth and partnership that have been honed by hundreds of millions of years of existence. Destroy a forest, and in 10 years, the fungal community is beginning to rebuild. In a century, the forest is returning. In 1,000 years, it approaches old growth complexity again. Not quickly, not without loss, but the direction is clear. Given time, given the chance, the fungi rebuild. There is one more thing to hold tonight, something that ties together everything we have spoken of across these many hours. Fungi do not know what they are doing. They do not plan. They do not intend. They do not hope. They simply grow, following the gradients of chemistry, reaching toward the food, branching when they find it, connecting when they meet compatible partners, doing what they have always done, and in doing so, without knowledge or intention or strategy, they sustain the forests. They build the soil. They heal the contaminated ground. They connect the trees. They cycle the carbon. They break the wood. They feed the seedlings. They shelter 300 species around the roots of a single ancient tree. They have been doing this for 1 billion years, and they will continue long after any particular forest has changed, long after any particular species has come and gone. The fungi will be here, quietly working in the dark beneath the floor of whatever world remains. That is a thought worth resting in tonight. Not a heavy thought, not a frightening thought, but a grounding one. A deep, slow, patient kind of hope. The kind that does not demand anything of you, that does not require you to act or worry or plan, only to rest, to breathe, to know that beneath the ordinary world, in the dark and the damp and the ancient soil, something extraordinary is at work. Something that has always been at work and always will be. The forest floor is alive tonight, as it has been every night since before the first tree stood. The mycelium is growing. The carbon is flowing. The spores are drifting on the dark air. The partnerships are forming. The old wood is becoming soil. The seedlings are finding the network. The fungi are holding it all together, quietly, without ceasing, without asking for anything in return. Let yourself be held by that tonight. You are resting in a world that knows how to heal itself. A world that has been doing it for longer than we can imagine, and will go on doing it long into a future that the fungi are already preparing. You are still here, still resting, still breathing slowly in the dark. The forest floor is all around you. The leaves, the soil, the pale threads of mycelium running in every direction beneath the surface. We have spent a long time in the company of fungi tonight, learning their ways, following their threads. And now, at the edge of that kingdom, right at the boundary where fungi give way to something stranger and older and harder to name, there lives a creature that does not quite belong anywhere. something that has puzzled naturalists for centuries, something that moves, that thinks in its own quiet way, that is alive in a manner quite unlike anything else on the forest floor, the slime mold. The name is not flattering. Slime mold. It conjures something unpleasant, something to step around. But set the name aside for a moment and simply look, because what you find when you actually look at a slime mold is something extraordinary, something almost impossibly beautiful, something that raises profound questions about the nature of life itself. Slime molds are not fungi. They were once classified as fungi. For a long time, they lived in the same taxonomic category, grouped together by their apparent similarities, both found on the forest floor, both often growing on decaying wood, both producing spores. But slime molds are, in their fundamental biology, something entirely different. They are now classified within a group called the amoebozoa, closer to amoebae than to fungi, closer in some ways to the ancestors of all complex life. They sit at the edge of the fungal world, not quite fungi, not quite anything else, their own ancient, strange, beautiful category. There are two main types of slime mold: the plasmodial slime molds and the cellular slime molds. They look similar from a distance, but their biology is fascinatingly different. The plasmodial slime molds form what is called a plasmodium, a single cell with many, many nuclei inside it, not a colony of cells working together, not a community. One cell, one continuous membrane, one cytoplasm, but containing potentially millions of nuclei. This single cell can grow quite large, spreading across the forest floor, branching and flowing, a network of tubes visible to the naked eye, pulsing and contracting, moving. A plasmodium flows across the surface it inhabits, moving at speeds of up to 4 centimeters per hour. That sounds slow, and it is. But for a single cell navigating a forest floor of leaf litter and bark and irregular surfaces, it is purposeful, directed, going somewhere where it is going is toward food. Slime molds feed on bacteria, on fungal spores, on the organic particles of the forest floor. As the plasmodium moves, it engulfs its food, surrounds it, digests it within the single vast cell. And it does not move randomly. It moves intelligently, or at least in a way that produces outcomes indistinguishable from intelligence. The most famous experiment in slime mold science was conducted in Japan in the early 2000s. A researcher named Toshiyuki Nakagaki placed a plasmodium of the yellow slime mold Physarum polycephalum at the entrance of a plastic maze. Oats were placed at the exit, and the slime mold was left to find them. 4 hours later, it had solved the maze, not by luck, not by filling every pathway and finding the food by chance, but by systematically retracting from dead ends and reinforcing the shortest, most efficient route between entrance and exit. The route it built was the optimal path. A single cell with no brain, no nervous system, no eyes, had solved a spatial problem that would require conscious reasoning from a human. Even more remarkable was a later experiment. Researchers mapped the major cities of Japan on a flat surface, placing food sources at the positions of the cities, and they introduced Physarum at the position of Tokyo. They let it grow and watched as it spread outward, exploring, connecting the food sources, building a network, and the network it built, the efficient, redundant, resilient network of tubes connecting city to city, closely resembled the actual rail network of Japan. The Shinkansen, the bullet train routes, the system that teams of engineers had spent decades designing, the slime mold in a matter of days, had independently arrived at something close to the same solution, using no intelligence, no planning, no tools, only chemistry, and the patient, distributed logic of a single flowing cell. How does it do this? The answer lies in the tubes. The plasmodium is not a featureless blob. It is a network of tubes. Cytoplasm flows through these tubes, and the tubes reinforce themselves where flow is greatest. Tubes with strong flow grow thicker. Tubes with weak flow thin out, eventually disappear. This feedback loop, thick tubes where flow is strong, thin tubes where flow is weak, naturally produces efficient networks. Roots that carry resources well are reinforced. Roots that are dead ends are abandoned. No mind required. Only the physics of flow and a few 100 million years of evolution. The slime mold also has memory, not memory in the way we experience it. Not images or words or feelings recalled, but a functional, physical memory. As physarum moves across a surface, it leaves behind a trail of slime, not the living slime of the plasmodium itself, but a different substance, an extracellular slime that marks where the organism has already been. When the plasmodium encounters this old slime, it turns away. It has, in effect, left a note for itself. I have already been here. There is nothing new in this direction. Go somewhere else. An external memory, written in chemistry, on the surface of the forest floor. There is something deeply calming about this, the idea of a mind without a brain, of memory without neurons, of intelligence without intention. The slime mold does not know it is being intelligent. It does not experience the satisfaction of solving a maze. It simply flows, following the gradients of chemistry, responding to what it finds, withdrawing from what offers nothing, reinforcing what works, and in doing so, producing outcomes that we, with all our complexity, struggle to match. On the forest floor, slime molds are humble and beautiful things. You find them most often on the underside of logs, on the damp surfaces of fallen leaves, on the bark of rotting stumps. In their plasmodial stage, they look like a thin, pale, or yellow, or orange film spread across the surface, glistening slightly with moisture. Sometimes, if you watch very carefully and very patiently, you can see it moving, a slow, almost imperceptible flow. The whole organism gently shifting, reaching toward somewhere new. When conditions become unfavorable, when food runs out, or the surface dries, or light becomes too strong, the slime mold does something extraordinary. It transforms. The plasmodium, that single flowing cell, gathers itself, rises upward, and produces fruiting bodies, tiny, intricate structures, some no taller than a millimeter, some looking under a lens like miniature coral, or tiny jeweled cups, or pale spheres on fine stalks, each one containing spores, each spore a new beginning, waiting for conditions to improve, waiting as everything in the forest waits, with patient and unhurried readiness. The cellular slime molds are different again, and even stranger in some ways. They spend most of their lives as individual single cells, separate, each one hunting bacteria on its own, each one an independent organism. But when food runs out, when the environment becomes difficult, something changes. The individual cells release a chemical signal, a call, and the other cells respond. They begin to move toward the signal, streaming together, gathering from across the leaf litter. Thousands of individual cells coming together, aggregating into a single slug-like mass. This slug, made of thousands of distinct individuals, moves as one, a coordinated body, going somewhere, finding better conditions, and then transforming together into a fruiting body, some cells becoming the stalk, dying in the process so that others, the ones that will become spores, can be elevated above the leaf litter, carried further by the wind, given a better chance of finding somewhere new. Individuals sacrificing themselves for the collective, a slime mold making a choice that seems almost moral. Scientists are careful about the language of choice and sacrifice when describing non-conscious organisms, and rightly so. These are chemical processes, responses to gradients. There is no suffering, no intention, no awareness, and yet the outcome is the same. Some cells die so that others may disperse. The group survives. The individual does not. Whatever words we choose for it, the pattern is real, and it is ancient, older than any nervous system, older than any brain. The slime molds remind us quietly that intelligence and complexity can arise from the simplest of foundations, from chemistry, from flow, from the patient, distributed logic of cells responding to their environment. They sit at the edge of the fungal kingdom, not quite belonging, but deeply at home on the forest floor, moving slowly through the damp leaf litter, solving problems no one set for them, remembering without a mind, choosing without a will, simply being in their own extraordinary way, the most ancient kind of alive. The forest floor is full of these quiet wonders tonight, things that do not announce themselves, that do not ask to be noticed, that simply get on with the business of living, slowly, in the dark, in the damp spaces between the leaves. Let them be with you now, the slime mold, flowing in its unhurried way across the surface of an old log, the mycelium branching in the soil, the spores drifting on the cool air, all of them alive in their own particular way, all of them part of the same vast, quiet, extraordinary world. Rest now. The forest is alive all around you. Every surface, every grain of soil, every fallen leaf, alive with something patient and strange and beautiful, and you resting within it all. Still here, still breathing. The night is very quiet now. Let your body be completely still. Let your thoughts be loose and soft. The forest around you is peaceful tonight, cool and dark and deeply, deeply alive. But forests have not always been peaceful. They have known fire. They have always known fire. And tonight, we are going to sit with the story of what happens after. After the flames have passed, after the smoke has cleared, after the ash has settled on the forest floor like a gray, soft snow. Because what happens after fire is one of the most remarkable stories in all of the natural world. And the fungi are at the very center of it. Fire is not new to forests. It has been part of the forest cycle for as long as forests have existed, long before any human ever struck a spark. Lightning started fires. Volcanic activity started fires. The dry heat of long summers started fires. And forests, over hundreds of millions of years, have evolved not merely to survive fire, but in some cases to depend on it, to incorporate it into the rhythm of renewal, the way a forest incorporates the death of a tree or the fall of a leaf. Fire, in the long view, is part of how a forest breathes. In the immediate aftermath of a fire, the forest floor looks like devastation. The leaf litter is gone. The mosses and small plants are gone. The surface of the soil is bare and gray. The fallen logs are charred. The standing trees, if they survived at all, are scorched black to a height of several meters. The air smells of cold ash, and the soil, which was so full of life, so threaded with mycelium, so dense with bacteria and mites and springtails, and all the small creatures of the duff layer, is changed. The heat has killed many things in the most severely burned areas. The surface soil may have lost most of its fungal community, most of its microbial life. The threads of mycelium, so finely built over decades, have been destroyed. And yet, within days of the fire's passing, something begins to stir, something that was waiting, something that fire did not destroy, because not all fungi are equal in the face of fire. Some are killed by it, and some, extraordinary, ancient, improbable as it sounds, were made for it. These are the pyrophilous fungi, from the Greek per, meaning fire, and philos, meaning loving, the fire-loving fungi, species that have evolved specifically to colonize burned ground, that germinate only after fire, that require the chemical changes that fire leaves behind in order to grow. Some of them cannot grow anywhere else. The charred wood, the altered soil chemistry, the reduction of competition from other organisms. These are not obstacles for the pyrophilous fungi. They are invitations. Among the most beautiful and most celebrated of these fire followers is the morel, Morchella, that extraordinary mushroom with its honeycomb cap, its deeply pitted, ridged surface, like a brain made of caramel-colored velvet. Morels appear after forest fires in extraordinary abundance. In the spring following a burn, foragers travel from great distances to burned forests, knowing that in the ash and the char, morels will be pushing through the soil, sometimes in numbers almost beyond belief. Dense carpets of them across the burned ground, each one emerging from soil that a few months earlier was nothing but gray ash. Why morels fruit so abundantly after fire is still not fully understood. Scientists suspect several factors are at work. The fire kills competing fungi and bacteria that would otherwise suppress morel growth. The ash adds minerals to the soil. The heat may break the dormancy of morel spores that have been waiting in the soil for years, perhaps for decades. And the trees, stressed or killed by the fire, may send distress signals through the soil, chemical cues that trigger the morel mycelium to fruit, to reproduce quickly before the window of opportunity closes. The morel, sensing the disruption of the forest, blooms in the aftermath, a brief, abundant, beautiful response to destruction. Other pyrophilous fungi are less familiar, but no less remarkable. Bioluminescent highlandensis, a small, tawny mushroom that appears on burned soil and charred wood almost exclusively. If you find Bioluminescent highlandensis, you are standing on ground that has burned. It is the fungus that marks the place of fire, a living record of something that happened here. Rhizopogon species, truffle-like fungi, whose spores survive extreme heat and germinate readily in burned soil, begin to form mycorrhizal connections with the first tree seedlings that colonize the ash before any other fungi have returned, before the soil has recovered its complexity. The fire fungi are already there, already connecting, already preparing the ground for what comes next. This is the extraordinary thing about pyrophilous fungi. They are not simply survivors of fire. They are the first responders, the pioneers, the organisms that arrive in the burned landscape and begin immediately the slow work of rebuilding. They stabilize the ash, their hyphae thread through the bare, unstructured, burned soil, binding the particles, preventing the ash from being blown away by wind or washed away by rain. They begin the slow reconstruction of the soil food web, providing food and habitat for the first bacteria, the first mites, the first springtails returning from the edges of the burn. They form mycorrhizal partnerships with the first seedlings, giving those fragile new plants access to nutrients in a soil that has been stripped of almost everything. The fire fungi make it possible for the forest to begin again in the soil beneath a burned forest. Even where surface life has been destroyed, something important often survives. The spore bank, deep in the soil, below the layers most affected by the heat of the fire, fungal spores persist. Spores of mycorrhizal species, spores of decomposers, spores of the pyrophilous specialists, protected by the insulating properties of the soil above them, waiting in the dark, as spores always wait for conditions to change, for the moment to arrive. And when the fire has passed, and the ash has cooled, and the first rains come, they germinate, pushing new hyphae up through the burned soil, threading through the ash, beginning again. Researchers studying the recovery of fungal communities after severe forest fires in British Columbia found something that surprised them. Within 2 years of a fire, the diversity of fungal species in the burned soil was already showing signs of recovery, not the same community as before. That takes much longer, decades at least, in some cases over a century, but a functional community, a working community, decomposers breaking down the charred wood. It does not rebuild from the top down. It rebuilds from below. The fungi come first, before the mosses, before the wildflowers, before the first tree seedlings. The fungi arrive in the ash. They prepare the soil. They make it possible for everything else to follow. The same pattern repeated across geological time. The fungi preparing the ground for the plants, as they did 500 million years ago when the land was bare rock, and the first plants were attempting their first tentative crossing from the water. The story of forest recovery after fire is, in miniature, the story of life on land. The fungi going first, making the world habitable, one slow thread at a time. Some forest ecologists speak of fire as a reset, a disturbance that clears the accumulated complexity of decades, opens the canopy to light, returns nutrients locked in old wood back to the soil, creates a mosaic of burned and unburned areas. Burned areas with their pyrophilous fungi and their pioneer seedlings. Unburned areas with their intact networks and their old trees, the two together creating a richer, more diverse landscape than either alone. The forest that grows back after fire is not the same forest that burned, but it is not lesser. It is different, shaped by fire in ways that may make it, eventually, more resilient, more complex, more alive. Tonight, the forest around you is untouched by fire. It is cool and still and dark. The leaf litter is deep and soft. The mycelium is intact. The networks are running their slow, quiet signals from tree to tree. But somewhere, not too far away, a burn scar from last summer's fire is quiet under the stars. The ash is cold. The charred logs are still, and in the soil beneath the ash, in the dark, below the reach of the fire's heat, spores are waiting, patient and intact and ready. The fire fungi sleeping, waiting for spring, waiting for the rain, waiting for the moment when the forest will begin again. It always begins again. It always has. Let that patience carry you into sleep now. The forest knows how to rebuild. It has been doing it longer than we have been watching. Rest now. Everything returns. Still breathing. Still here. The night is soft around you, deep and unhurried. We have spent so much time tonight in the invisible world, in the mycelium beneath the soil, in the networks threading through the roots, in the chemistry of glomalin and spores and bioluminescence. But tonight, for a while, we are going to slow down even further and simply look at a mushroom. One single mushroom, standing in the quiet of the forest floor, and take our time with it. Because the mushroom, that familiar thing we pass without much thought, is a structure of extraordinary elegance. a temporary miracle built with great precision for a single, brief, essential purpose. Imagine you are kneeling on the forest floor. The soil is cool and damp beneath your knees. The light is low and golden. And there, pushing up through the leaf litter at the base of an old oak tree, a mushroom, freshly emerged, still young, the cap not yet fully open. The cap, in the language of mycology, the pileus, from the Latin word for hat. And that is exactly what it is. A hat, a dome of tissue held above the forest floor, sheltering what lies beneath. The cap of a young mushroom is closed, rounded, sometimes bullet-shaped, sometimes perfectly hemispherical, pulled tight around the stem, as a bud is tight before it opens. As it ages, it expands. The margin, the outer edge of the cap, lifts, the dome flattens, and eventually, in many species, the cap opens fully, flat or even upturned, a wide, shallow platform reaching out into the air, exposing the structures beneath. The color of the cap is one of the most variable things in the mushroom world. Pure white, deep blood red, pale lemon yellow, rich chestnut brown, spotted, striped, smooth as a dinner plate, rough with scales or warts or a powdery bloom. The enormous diversity of cap colors and textures is not random. Some colors are warnings. Bright reds and yellows often signal toxicity, a message to potential grazers. Something chemical and dangerous lives inside. Move on. Other caps are cryptic, brown and gray, and mottled to blend with the leaf litter, not calling attention, not warning, simply resting in the forest, quietly, unnoticed. Look now at the underside of the cap, and here is where the mushroom reveals its true purpose. In the most familiar mushrooms, the ones we recognize from fields and forest edges and supermarket shelves, the underside of the cap is lined with gills. The lamellae, thin, blade-like structures radiating outward from the center, from the attachment point at the stem, out toward the edge of the cap, like the spokes of a wheel, or the pages of a book held open. Each gill is a thin, vertical sheet of tissue, and across both surfaces of each gill, spores are produced. Millions of them, packed densely into the gill surface, waiting for the moment of release. The gill surface is lined with specialized cells called basidia. Each basidium produces exactly 4 spores. 4 in most species, held on tiny projections called sterigmata, like 4 children sitting on the tips of 4 fingers. When the spores are mature, they are forcibly discharged, catapulted from the basidium by a tiny drop of water that forms at the base of each spore. The surface tension of this drop snaps the spore free, launching it horizontally, a fraction of a millimeter, just far enough to clear the gill surface. And then it falls straight down through the narrow channel between adjacent gills, out from under the cap, into the air and away. The spacing of the gills is precise, not random, not incidental. The distance between adjacent gills is wide enough for spores to fall freely without hitting the opposite gill, and narrow enough to maximize the total gill surface area beneath the cap. The mushroom has solved an engineering problem. More surface area means more spores. But if the gills are too close together, the spores cannot escape. The precise spacing of the gills is the elegant solution, arrived at not by calculation, but by natural selection over millions of years. The mushrooms that packed their gills too closely produced fewer surviving spores. The ones that found the optimum spacing reproduced more successfully, and the spacing we see today in every gilled mushroom in every forest on Earth is the result. Not all mushrooms have gills. Some have pores. The bolete, that group of sturdy, thick-stemmed mushrooms that includes the famous porcini, the cep, Boletus edulis, beloved of foragers across Europe and North America. Instead of gills, the underside of a bolete's cap is a dense sponge of tiny tubes, each tube opening at the base of the cap in a small round pore. The spores are produced inside the tubes and fall from the openings. The effect from below is like looking up at a honeycomb. Thousands thousands of tiny openings, each one a portal through which the next generation passes. And some mushrooms have teeth, downward pointing spines, like a soft, white stalactite cave. Lion's mane, which we spoke of in our section on healing, is the most dramatic example. Its cascading white spines, each producing spores along their length. A waterfall of spore-bearing tissue, extraordinary to see. Now let us descend to the stem, the stipe, that upright column that holds the cap above the forest floor. The stem is not merely a support structure. It is plumbing. Water and nutrients flow upward through the stem from the mycelium below. The cells of the stem are aligned vertically, long and parallel, arranged for the efficient transport of fluid, like the grain of wood, like the fibers of a muscle. In some species, the stem is solid, dense, and firm throughout. In others, it is hollow, a tube of tissue surrounding an empty central channel. In others still, it is stuffed with a loose, cottony tissue, soft and white and dry. Near the top of the stem, in many species, you will find a ring, the annulus, a skirt of pale tissue hanging from the stem. Fragile, sometimes disappearing with age, the annulus is a remnant, a ghost of something that existed when the mushroom was young, when the cap was still closed and the gills were still hidden. The entire underside of the cap was covered by a thin membrane. The partial veil stretching from the edge of the cap to the upper part of the stem, protecting the gills during the mushroom's early development, keeping them safe while the spores matured. But as the cap expanded, as the mushroom grew and the dome opened, the partial veil stretched and tore, and what remained clinging to the stem became the ring, a record of the cap's opening, a mark of the mushroom's transition from young to mature. At the very base of the stem, in some species, there is another remnant, the vulva, a cup of tissue at the stem's base, partly buried in the soil. The vulva is all that remains of an earlier membrane, one that covered not just the gills, but the entire mushroom. The universal veil before the mushroom emerged from the soil. The universal veil enclosed it completely. like an egg, like a tight, protective wrapping. And as the mushroom pushed upward through the soil, as its stem lengthened and its cap rose, the universal veil ruptured, leaving the vulva at the base, and sometimes scattered across the cap's surface, pale patches or warts, the remaining fragments of that original protective membrane. The Amanita mushrooms, that famous and beautiful and often deadly family, are the classic example. The death cap, the destroying angel, both enclosed in a universal veil when young, both bearing a vulva at the base when mature. The vulva is, for the mushroom forager, one of the most important things to check. Its presence is a signal. Look carefully. Check every feature. This mushroom may be beautiful and dangerous, but set the toxicology aside for tonight. We are simply looking. Simply being with this single mushroom on the forest floor, taking in its form the closed dome of the young cap, the expanding, opening disc of the mature one, the precise spacing of the gills beneath, the smooth or shaggy or scaled surface above, the ring halfway up the stem, the vulva at the base, and within all of it, the slow, patient work of producing spores, billions of them, released in a slow, continuous fall. Through the night air, the mushroom will not last long. Its entire above-ground existence spans days, a week, perhaps, for the largest and most robust. Many last only hours before the insects find them, before the slugs and the deer mice and the forest voles take their share, before the heat or the rain or the frost ends the brief flowering of the fruiting body. But in that brief time, it has done its work. The spores have been released, billions of them, drifting on the forest air, each one carrying the potential for a new network, a new forest, a new partnership. The mushroom does not know any of this. It does not experience its brief existence. It does not mourn its passing. It simply grows, opens, releases, and returns. The mycelium beneath, patient and intact, has already begun preparing the next fruiting body, the next brief rising, the next small, perfect, temporary miracle, and another, until the mycelium itself returns to the soil. And from the soil, through the slow chemistry of decay, the elements of a new fungus are already forming. The cycle turning quietly, without end. Rest now. The mushroom on the forest floor is releasing its spores into the cool night air. Billions of them drifting upward in the stillness, rising past the leaf litter, past the moss, into the dark space beneath the trees, carried away on the faintest movement of air. Each one going somewhere, each one a small, patient beginning. Let your thoughts be like those spores tonight, drifting, weightless, going somewhere without effort, without intention, simply carried into the deep, soft dark, still breathing, still here. The night is soft around you, deep and unhurried. We have spent so much time tonight in the invisible world, in the mycelium beneath the soil, in the networks threading through the roots, in the chemistry of glomalin and spores and bioluminescence. But tonight, for a while, we are going to slow down even further and simply look at a mushroom, one single mushroom, standing in the quiet of the forest floor, and take our time with it. Because the mushroom, that familiar thing we pass without much thought, is a structure of extraordinary elegance, a temporary miracle, built with great precision for a single, brief, essential purpose. Imagine you are kneeling on the forest floor. The soil is cool and damp beneath your knees. The light is low and golden. And there, pushing up through the leaf litter at the base of an old oak tree, a mushroom, freshly emerged, still young, the cap not yet fully open. The cap, in the language of mycology, the pileus, from the Latin word for hat. And that is exactly what it is. A hat, a dome of tissue held above the forest floor, sheltering what lies beneath. The cap of a young mushroom is closed, rounded, sometimes bullet-shaped, sometimes perfectly hemispherical, pulled tight around the stem, as a bud is tight before it opens. As it ages, it expands. The margin, the outer edge of the cap, lifts, the dome flattens, and eventually, in many species, the cap opens fully, flat or even upturned, a wide, shallow platform reaching out into the air, exposing the structures beneath. The color of the cap is one of the most variable things in the mushroom world. Pure white, deep blood red, pale lemon yellow, rich chestnut brown, spotted, striped, smooth as a dinner plate, rough with scales or warts or a powdery bloom. The enormous diversity of cap colors and textures is not random. Some colors are warnings. Bright reds and yellows often signal toxicity, a message to potential grazers. Something chemical and dangerous lives inside. Move on. Other caps are cryptic, brown and gray, and mottled to blend with the leaf litter, not calling attention, not warning, simply resting in the forest, quietly, unnoticed. Look now at the underside of the cap, and here is where the mushroom reveals its true purpose. In the most familiar mushrooms, the ones we recognize from fields and forest edges and supermarket shelves, the underside of the cap is lined with gills. The lamellae, thin, blade-like structures radiating outward from the center, from the attachment point at the stem, out toward the edge of the cap, like the spokes of a wheel, or the pages of a book held open. Each gill is a thin, vertical sheet of tissue, and across both surfaces of each gill, spores are produced. Millions of them, packed densely into the gill surface, waiting for the moment of release. The gill surface is lined with specialized cells called basidia. Each basidium produces exactly 4 spores. 4 in most species, held on tiny projections called sterigmata, like 4 children sitting on the tips of 4 fingers. When the spores are mature, they are forcibly discharged, catapulted from the basidium by a tiny drop of water that forms at the base of each spore. The surface tension of this drop snaps the spore free, launching it horizontally, a fraction of a millimeter, just far enough to clear the gill surface. And then it falls straight down through the narrow channel between adjacent gills, out from under the cap, into the air and away. The spacing of the gills is precise, not random, not incidental. The distance between adjacent gills is wide enough for spores to fall freely without hitting the opposite gill, and narrow enough to maximize the total gill surface area beneath the cap. The mushroom has solved an engineering problem. More surface area means more spores. But if the gills are too close together, the spores cannot escape. The precise spacing of the gills is the elegant solution, arrived at not by calculation, but by natural selection over millions of years. The mushrooms that packed their gills too closely produced fewer surviving spores. The ones that found the optimum spacing reproduced more successfully, and the spacing we see today in every gilled mushroom in every forest on Earth is the result. Not all mushrooms have gills. Some have pores. The bollites, that group of sturdy, thick-stemmed mushrooms that includes the famous porcini, the cep, Boletus edulis, beloved of foragers across Europe and North America. Instead of gills, the underside of a bolite's cap is a dense sponge of tiny tubes, each tube opening at the base of the cap in a small round pore. The spores are produced inside the tubes and fall from the openings. The effect from below is like looking up at a honeycomb. Thousands of tiny openings, each one a portal through which the next generation passes, and some mushrooms have teeth, downward pointing spines like a soft, white stalactite cave. Lion's mane, which we spoke of in our section on healing, is the most dramatic example. Its cascading white spines, each producing spores along their length. A waterfall of spore-bearing tissue, extraordinary to see. Now let us descend to the stem, the stipe, that upright column that holds the cap above the forest floor. The stem is not merely a support structure. It is plumbing. Water and nutrients flow upward through the stem from the mycelium below. The cells of the stem are aligned vertically, long and parallel, arranged for the efficient transport of fluid, like the grain of wood, like the fibers of a muscle. In some species, the stem is solid, dense, and firm throughout. In others, it is hollow, a tube of tissue surrounding an empty central channel. In others still, it is stuffed with a loose, cottony tissue, soft and white and dry. Near the top of the stem, in many species, you will find a ring, the annulus, a skirt of pale tissue hanging from the stem. Fragile, sometimes disappearing with age, the annulus is a remnant, a ghost of something that existed when the mushroom was young, when the cap was still closed and the gills were still hidden. The entire underside of the cap was covered by a thin membrane. The partial veil, stretching from the edge of the cap to the upper part of the stem, protecting the gills during the mushroom's early development, keeping them safe while the spores matured. But as the cap expanded, as the mushroom grew and the dome opened, the partial veil stretched and tore, and what remained clinging to the stem became the ring, a record of the cap's opening, a mark of the mushroom's transition from young to mature. At the very base of the stem, in some species, there is another remnant, the vulva, a cup of tissue at the stem's base, partly buried in the soil. The vulva is all that remains of an earlier membrane, one that covered not just the gills, but the entire mushroom. The universal veil before the mushroom emerged from the soil. The universal veil enclosed it completely, like an egg, like a tight, protective wrapping. And as the mushroom pushed upward through the soil, as its stem lengthened and its cap rose, the universal veil ruptured, leaving the vulva at the base, and sometimes scattered across the cap's surface, pale patches or warts, the remaining fragments of that original protective membrane. The Amanita mushrooms, that famous and beautiful and often deadly family, are the classic example. The death cap, the destroying angel, both enclosed in a universal veil when young, both bearing a vulva at the base when mature. The vulva is, for the mushroom forager, one of the most important things to check. Its presence is a signal. Look carefully. Check every feature. This mushroom may be beautiful and dangerous, but set the toxicology aside for tonight. We are simply looking. Simply being with this single mushroom on the forest floor taking in its form the closed dome of the young cap, the expanding, opening disc of the mature one, the precise spacing of the gills beneath, the smooth or shaggy or scaled surface above, the ring halfway up the stem, the vulva at the base, and within all of it, the slow, patient work of producing spores, billions of them, released in a slow, continuous fall. Through the night air, the mushroom will not last long. Its entire above-ground existence spans days, a week, perhaps, for the largest and most robust. Many last only hours before the insects find them, before the slugs and the deer mice and the forest voles take their share, before the heat or the rain or the frost ends the brief flowering of the fruiting body. But in that brief time, it has done its work. The spores have been released, billions of them, drifting on the forest air, each one carrying the potential for a new network, a new forest, a new partnership. The mushroom does not know any of this. It does not experience its brief existence. It does not mourn its passing. It simply grows, opens, releases, and returns. The mycelium beneath, patient and intact, has already begun preparing the next fruiting body, the next brief rising, the next small, perfect, temporary miracle, and another, until the mycelium itself returns to the soil. And from the soil, through the slow chemistry of decay, the elements of a new fungus are already forming. The cycle turning quietly, without end. Rest now. The mushroom on the forest floor is releasing its spores into the cool night air. Billions of them drifting upward in the stillness, rising past the leaf litter, past the moss, into the dark space beneath the trees, carried away on the faintest movement of air, each one going somewhere, each one a small, patient beginning. Let your thoughts be like those spores tonight, drifting, weightless, going somewhere without effort, without intention, simply carried into the deep, soft dark. Let your breathing follow the rhythm of water tonight, not rushing, not turbulent, just the steady, soft movement of a shallow stream. running over smooth stones in the dark, catching no light, making the gentlest of sounds. The forest tonight has water in it, a stream threading through the trees, a bog at the far edge of the wood, dark and still and very ancient. And in both places, in the water and in the peat, the fungi are present, doing what they have always done, quietly, completely. We tend to think of fungi as creatures of the soil, of the forest floor, of the dry, open air where spores can drift. But fungi have colonized water too. They have found ways to live in streams and rivers and bogs and wetlands. They have adapted to the particular challenges of an aquatic life. And in doing so, they have become essential to the health of every freshwater ecosystem on Earth. The aquatic hyphomycetes are among the least known and most important fungi in the forest. Their name is not familiar. They do not produce mushrooms. They do not glow in the dark. They are invisible to any ordinary observer, but in the cold, clear, fast-flowing streams that run through temperate forests, they are the dominant decomposers, the primary agents of transformation, the organisms most responsible for breaking down the leaves that fall into the water each autumn and turning them into something the stream ecosystem can use. Each autumn, when the leaves fall, enormous quantities of leaf litter enter forest streams. Thousands of tons of organic material dropped into the water from the overhanging branches, settling on the stream bed, accumulating in the slow pools and the sheltered bends. This leaf material is rich in carbon, rich in energy, but it is also tough, difficult to digest, full of lignin and cellulose and tannins and waxes, materials that resist breakdown, that would persist in the water for a very long time without the right organisms to process them. The aquatic hyphomycetes are those organisms within days of a leaf entering a forest stream. The aquatic hyphomycetes have found it. Their spores, drifting in the current, settle on the leaf surface, germinate, begin to grow hyphae through the leaf tissue, producing the same powerful enzymes that their terrestrial cousins use on the forest floor. cellulases, ligninases, breaking down the tough structural compounds, softening the leaf, making it more digestible, more nutritious, more palatable. But the aquatic hyphomycetes are not only breaking down leaves for their own benefit, they are transforming them for the entire stream community. As the fungal hyphae grow through a leaf, they become incorporated into the leaf tissue. The leaf, now colonized by fungal mycelium, is significantly richer in protein and nitrogen than the original leaf. The fungal biomass has improved the leaf's nutritional value, and the stream insects, the caddisfly larvae, the stonefly nymphs, the mayfly larvae, prefer colonized leaves. They select them deliberately, choosing the fungus-colonized material over uncolonized leaves. The fungi, in processing the leaves, have made them into food, not just for themselves, but for the invertebrates, who are in turn food for the fish, who are in turn food for the kingfisher, the otter, the heron, the fungi, invisible on the stream bed, supporting the entire food web of the living stream. The spores of aquatic hyphomycetes are among the most beautiful structures in mycology, where terrestrial fungi produce spores that are round or oval or simple, shaped for drifting in air. The aquatic hyphomycetes produce spores of extraordinary architecture: four-armed stars, curved commas, branched filaments, spirals, shapes that under a microscope look like something a jeweler might design. These elaborate shapes are not decorative. They are functional. In flowing water, a round spore will roll and tumble and be carried away. A four-armed star, by contrast, catches on surfaces, lodges in the spaces between particles, hooks onto the substrate, anchors itself against the current. The elaborate shape of the aquatic hyphomycete spore is an adaptation to life in water, a solution to the problem of staying in place when everything is moving. Some aquatic hyphomycetes lead a double life. They spend part of their existence in the stream and part of it in the forest above, as endophytes living inside the leaves of trees while those leaves are still alive, dormant, causing no apparent harm, waiting when those leaves fall into the stream, the fungi within them are already present, already positioned, ready to begin the decomposition process from inside the leaf tissue. Before any external spore has even had time to settle. A quietly brilliant strategy, embedding in the leaf before it falls, traveling from the canopy to the stream bed within the leaf itself, arriving already in place. Now let us move from the stream to the bog, a different kind of water, still, dark, ancient. A raised bog on a cool, cloudy day is one of the strangest and most beautiful landscapes in the world. The surface is soft and spongy underfoot. The air smells of peat and cold water. The vegetation is low and windswept, heathers and sedges, and the extraordinary sphagnum moss, that great, water-holding, world-shaping plant, pale green and russet and deep burgundy, growing in cushions and hummocks across the bog surface. Sphagnum moss does something remarkable. It creates its own environment. As it grows, it acidifies the water around it, taking up minerals from the rainwater and releasing hydrogen ions, making the bog water so acid that almost nothing can decompose in it. This is why bogs preserve things so well. The famous bog bodies, thousands of years old, their skin still intact, their features still recognizable. The Toland man, the Lindo man, preserved by the cold, acid, airless water of the bog, the conditions that prevent almost all microbial activity, that hold things exactly as they were, for centuries, for millennia. But even in this inhospitable environment, the fungi are present, working in their patient, adapted way. Some fungal species have evolved to tolerate the extreme acidity of bog water. Others work at the aerobic margins of the bog, where the sphagnum surface meets the air, where some oxygen is still available, and in these marginal zones, a slow, careful decomposition takes place. Not fast, not the rapid turnover of the forest floor, but a decomposition calibrated to the limits of the environment, producing over centuries the layered, compressed organic material of peat. peat is essentially incompletely decomposed plant material. Sphagnum moss and sedges and heathers and all the other bog plants, built up over thousands of years at the rate of about 1 millimeter per year. The same patience as the lichen on the Arctic rock, the same slow accumulation, layer upon layer, season upon season. In some bogs, the peat is 10 meters deep, 10,000 years of slow accumulation. 10,000 years of the fungi working at the margins, breaking down what they can, leaving the rest to the cold and the acid, preserved. The carbon stored in the world's peatlands is extraordinary, though peatlands cover only about 3% of the Earth's land surface. They store approximately twice as much carbon as all the world's forests combined. The ancient, cold, acid, slow decomposing peat holding the carbon of 10,000 years of plant growth, locked away, held by the very conditions that make rapid fungal decomposition impossible. The bog, in its stillness and its cold and its acid, is one of the great carbon vaults of the earth. And the fungi, paradoxically, are part of why their limited activity in the bog conditions is precisely what allows the peat to accumulate, what allows the carbon to stay. By the stream tonight, the aquatic hyphomycetes are at work, their branched and star-shaped spores drifting in the current, settling on the autumn leaves, beginning the slow, essential work of transformation. The caddisfly larvae are browsing the soft, colonized leaf material. The trout, somewhere deeper in the pool, are resting in the cold, dark water. And at the edge of the bog, the sphagnum is holding the rain. That extraordinary moss, absorbing 20 times its own weight in water, building the peat millimeter by millimeter, year by year, water and fungi, two of the quietest, most patient things in the natural world, moving together, shaping the stream, preserving the bog, sustaining the food webs of every freshwater place, in the dark, under the surface, where no one looks, where the work has always been done. Let the sound of the stream carry you now. That soft, unhurried movement of water over stone, cool and dark and clear. The fungi are in it. The fungi are always in it. Everything connected, everything held. Rest now. The water is moving. The bog is still. And somewhere between the two, the forest breathes. Breathe in slowly, deeply. Imagine the air of the forest coming into you now. Cool, damp, rich with something that has no easy name. Something that reaches into the deepest, oldest part of the brain. Something that feels immediately like home, like safety, like the beginning of rest. The smell of the forest after rain. Tonight, we follow that smell. We trace it back to its source, down through the air, through the leaf litter, into the soil, into the ancient chemistry of bacteria and fungi, and the long, slow work of the earth. The smell has a name: petrichor. Coined in 1964 by two Australian scientists, Isabel Joy Baer and Richard Thomas, from the Greek petra, meaning stone, and ichor, the mythical fluid that flows in the veins of the gods, the fluid of the stone, the essence released when rain falls on dry earth. It is one of the most universally recognized and universally loved of all natural scents. People who have lived their whole lives in cities know it. People who have never walked in a forest know it. It rises from pavements, from garden soil, from the dust of country roads. Wherever the earth has been dry and rain falls, that smell rises. Petrichor is not a single compound. It is a mixture, a complex, layered blend of volatile molecules released from soil and plant and fungal matter when raindrops disturb the surface. The first component to be identified was a group of plant oils, compounds produced by vegetation during dry periods, stored in the soil surface, released by rain. These oils contribute a green, waxy note, a faint suggestion of living plant matter, but the most powerful and most characteristic component of petrichor is something else. Something produced not by plants, but by microbes. Geosmin, geo, meaning earth. Osmin, from the Greek osmi, meaning smell. The smell of the earth, a single molecule. a bicyclic alcohol built from carbon and hydrogen and oxygen, with an earthy, musty, deeply rich quality, and produced primarily by a group of soil bacteria called actinomycetes, particularly the genus Streptomyces. These bacteria are among the most important in the soil food web. They are decomposers. They are producers of antibiotics. Many of our most important medicines, including streptomycin, the first antibiotic effective against tuberculosis, were derived from streptomyces, and they produce geosmin continuously as part of their ordinary metabolism, released when they die, released when rain disrupts the soil surface, released into the air we breathe. Human beings are extraordinarily sensitive to geosmin, among the most sensitive of any compound that our noses can detect. The threshold for smelling geosmin is approximately 5 parts per trillion. 5 parts per trillion. A single drop of geosmin dissolved in 200 Olympic swimming pools of water, and we would still smell it. Our noses, for reasons that scientists are still debating, are tuned with extraordinary precision to this particular molecule. Why? The most compelling explanation is evolutionary. Our ancestors, not the distant ones, but the relatively recent ones, lived in landscapes where water was precious, where finding water meant survival. And the smell of geosmin, rising from soil disturbed by rain, was one of the most reliable signals that water was nearby, that rain had fallen, that a pool or a stream or a patch of damp earth was close. The humans who could smell geosmin at the lowest concentrations were the ones most likely to find water in a dry landscape, the ones most likely to survive. And so our extraordinary sensitivity to this smell was selected for, preserved, passed down through 10,000 generations, until today, when you breathe in the smell of rain on earth and something ancient in you recognizes it, not intellectually, not by memory, but somewhere deeper, in the part of the brain that is older than thought. Fungi produce geosmin too, not as the primary producers. That role belongs belongs to the actinomycetes, but several fungal species contribute to the earthy scent of the soil, and fungi produce their own remarkable suite of volatile compounds. The characteristic mushroom smell, that deep, complex, slightly earthy, slightly nutty, slightly woody scent of a fresh mushroom, has a primary compound, one molecule most responsible for it, 1-octanthreol, sometimes called mushroom alcohol, an 8-carbon compound produced when fungi break down the fatty acids in their cell membranes, released as the fungal tissue ages or is damaged or begins to decompose. 1-octanthreol is not just a smell. It is a signal for many insects and small arthropods. It is one of the primary chemical cues for locating fungi. The compound drifts from the fruiting mushroom, and the insects that feed on fungal flesh follow the gradient, moving toward higher concentrations, finding the mushroom, and in finding it, helping to disperse the spores, carrying them away on their bodies into new parts of the forest. The smell of the mushroom is an invitation, a chemical broadcast saying to those with the sensory equipment to receive it, "Here, come here. There is something here for you." Different mushrooms produce different aromatic profiles. Some are extraordinary. The matsutake mushroom, so revered in Japanese cuisine that a single specimen can sell for hundreds of dollars, produces a scent unlike any other fungus. Sweet and spicy and piney and rich. Its primary aromatic compound is methylcinnamate, described as balsamic, strawberry, cinnamon, fruity and warm and deeply complex. A compound the matsutake produces in part to deter the organisms that would eat it before its spores are mature. The smell is both an invitation and a warning. Come to the insects that will spread its spores, but not yet, not until I am ready. The chanterelle smells of apricot, of warm fruit and mild pepper. The hedgehog mushroom, with its pale downward pointing teeth, smells faintly of wood and earth and something sweeter. The ink caps, those delicate mushrooms that dissolve into black ink as they mature, smell of ink and sea, and something almost metallic, each species its own fingerprint, its own particular arrangement of volatile molecules released into the forest air, drifting through the trees, a chemistry that is, in its own way, a kind of language, a conversation between the fungus and the forest, between the fungus and the animals, between the fungus and us. The forest itself, beyond the fungi, is saturated with scent. The trees produce terpenes, monoterpenes, and sesquiterpenes, and diterpenes, alpha-pinene, and limonene, and camphene. The compounds that give pine forests their sharp, resinous freshness, that give eucalyptus forests their cool, medicinal clarity. These terpenes are not simply pleasant. They are functional. They signal stress. They attract pollinators. They deter herbivores. They communicate between trees. And they influence the atmosphere itself. Forests emit enough terpenes that they contribute to cloud formation above them. The volatile compounds rising from the trees, forming tiny aerosol particles, seeds around which cloud droplets form, the forest, in a sense, making its own rain. And when that rain falls, when those clouds, partly seeded by the forest's own chemistry, release their water onto the leaf litter and the soil below, the petrichor rises, the geosmin is released. The 1-octanthreol drifts from the rain-dampened mushrooms. The terpenes are washed from the air and mix with the complex chemistry of the wet soil. And the smell that reaches you, the smell that reaches deep into the oldest part of your brain and says something wordless and immediate, is all of that chemistry, all of that biology, all of that ancient, reciprocal, interconnected conversation between the forest and the rain and the soil and the fungi, rising to your nose in a single, perfect, wordless moment. The smell of the forest. Breathe it in now. Imagine it. The cool, damp air after rain. The rich, deep, earthy smell of the forest floor. The faint sweetness of the mushrooms nearby. The sharp, clean note of pine from somewhere above. The ancient, memory-triggering smell of wet stone and living soil. Let it move through you, Let it reach that part of the brain that recognizes it without thinking. That says, without words, this is safe. This is alive. This is home. Rest now. The forest is breathing around you, its chemistry moving through the air, through your lungs, into your blood. Ancient molecules meeting ancient receptors. In the quiet, unhurried dark, everything for this moment is exactly as it should be. The night has its own rhythm, separate from the day, slower, deeper, more patient. The forest that was still and dappled in afternoon light is in darkness, a different place entirely. The temperature has dropped. The humidity has risen. The dew is beginning to settle on the leaf litter, on the caps of the mushrooms, on the pale threads of mycelium just visible at the surface of the soil. And in this cool, damp, dark world, the fungi come alive in ways that daylight never allows. Tonight, we are going to stay right here, in the dark, and pay attention to what the night does to the forest and to the fungi within it. Humidity is the key, the single environmental factor that most consistently governs fungal activity. During the day, the sun draws moisture from the forest floor, from the leaf litter, from the soil surface. The relative humidity at ground level fluctuates, rising in the shade, falling in patches of sunlight, drying the surface of mushroom caps, making spore release less efficient. But at night, everything changes. The sun is gone, the temperature drops, and the humidity rises steadily through the evening, reaching its peak in the darkest hours before dawn. This is when the forest floor is most fully saturated with moisture, most hospitable, most alive with fungal activity. Spore dispersal in forest canopies is greatest at night, and in the early hours of the morning, when humidity is highest and spore germination rates are most favorable. This is not a coincidence. It is a pattern so consistent, so universal across fungal species and forest types, that it speaks to something fundamental about the relationship between fungi and the night. The darkness is not a resting time for the fungal world. It is, in many ways, the most active time, the time most suited to the essential work of reproduction, of dispersal, of finding new places to grow. Some fungi have taken this relationship with the night even further. They have evolved internal clocks, biological timekeepers, circadian rhythms, the same 24-hour internal timing system that governs the sleep and waking of animals, the flowering of plants, the tide reading of coastal organisms. Fungi have it too. In Neurospora crassa, the bread mold that has been one of the primary model organisms of genetics research for nearly a century, the circadian clock was first characterized. Neurospora grows in bands across its culture medium, alternating bands of dense and sparse growth, a physical record of the fungus's internal rhythm, growing one way during what its clock perceives as day, and another way during what it perceives as night, even in total darkness, even in a laboratory with no external light cues. The rhythm persists. The internal clock keeps running, telling the fungus what time it is, even when no external signal says so. The bioluminescent fungi, those glowing species we explored in an earlier section of tonight's journey, have a particularly elegant relationship with the night. Most of them glow continuously, around the clock, their chemistry producing light regardless of the hour. But one species, a tropical mushroom called Neonothopanus gardneri, does something more precise, something that shows the circadian clock at work in a direct and beautiful way. This mushroom glows brightest at night, not because the darkness makes the glow more visible, though it does, but because the mushroom itself is timing its peak luminescence for the night hours. Its circadian clock regulates the production of luciferin and luciferase, the chemical components of its light, ramping them up as darkness falls, scaling back as dawn approaches. The mushroom is scheduling its glow, saving its light for when it will be seen, for when the nocturnal insects that will spread its spores are active and searching. A clock inside a fungus, counting the hours of the night. The fungi also time their spore release using their internal clocks. Different spore types are released at different times of day. Thin-walled spores, more vulnerable to ultraviolet radiation, are released preferentially at night when UV levels are 0, when they can drift through the dark air without damage. More robust, UV-resistant spores are released during the day, each type timed to the conditions that give it the best chance. The fungus, incapable of seeing the sky, incapable of feeling the sun directly, knows through its internal clock what time it is and adjusts its behavior accordingly. Whose adults carry fungal spores on their bodies from mushroom to mushroom. These insects are tuned to the chemistry of the night fungi. The volatile compounds released by ripening fruiting bodies in the dark hours, the 1-octan-3-ol we talked about, drifting on the cool night air, drawing the insects toward it, toward the mushroom, toward the spores. The fungi and their insect partners meet in the dark. The insects drawn by chemistry, the fungi releasing their most potent signals in the hours of highest humidity, the hours of greatest insect activity. A meeting arranged by millions of years of coevolution, taking place every night in forests around the world while almost no one watches. The dew that settles on the forest floor at night plays a role too. Fine droplets of water condensing on every surface, on the leaf litter, on the caps of mushrooms, on the threads of mycelium visible at the soil surface. This dew reactivates fungal growth that may have slowed in the drier hours of the afternoon. Hyphae that had paused in the heat extend again. Spores that had waited for moisture begin to germinate. The forest floor, barely visible in the dark, is alive with movement at this microscopic scale. More alive in some ways than in the bright hours of the day. There is something deeply appropriate about spending this night in the company of the fungi. Because the fungi, more than almost any other kingdom of life, are creatures of the night, of the dark, of the cool, damp, unhurried hours when the world is still. They do not need sunlight. They do not photosynthesize. They do not reach toward the light. They reach inward and downward, into the soil, into the wood, into the hidden spaces where the nutrients wait, and they do their best work in the dark, in the humidity, in the stillness, in exactly the conditions that you are resting in tonight. The night is their home, and for these hours, it is yours too. The forest floor is dark and damp and full of slow activity, the mycelium extending millimeter by millimeter through the cool soil, the spores drifting on the night air, the bioluminescent mushrooms glowing softly at their peak, their circadian clocks telling them this is the moment. This is the deepest dark. This is the time. Everything timed, everything paced, everything belonging to the night, and you resting within it, breathing with the same slow rhythm, belonging here too. There is a conversation happening tonight, right now, in the soil beneath the forest floor, in the air between the trees, in the thin films of water coating every soil particle. A conversation with no words, no sound, no movement that any eye could detect, but a conversation nonetheless, complex, layered, reaching across distances both vast and microscopic. The fungi are talking to each other, to the trees, to the bacteria in the soil, to the insects moving through the leaf litter above. And tonight, very quietly, we are going to listen. Communication in the human sense requires a sender and a receiver, a signal and a response, intent, perhaps, or at least the appearance of it. The fungi have none of the structures we associate with communication. No brain, no nervous system, no vocal cords or hands or eyes. And yet the signals they send and receive are real. The responses they produce are measurable. The outcomes of their chemical conversations shape the forest in ways we are only beginning to understand. The primary language of the fungal world is chemistry. Volatile organic compounds, molecules small enough to evaporate, to diffuse through air and water and soil, to travel from one organism to another, carrying information encoded not in symbols or sounds, but in molecular shape, in the particular geometry of an atom arrangement that a receptor somewhere is perfectly shaped to receive. When a mycorrhizal fungus encounters the root of a compatible tree for the first time, it does not simply attach itself. It sends a signal first, a chemical greeting, a set of volatile compounds that say, in the language of chemistry, I am here. I am a certain kind of fungus. Are you a compatible host? And the root responds with its own set of compounds, saying something back, something that either welcomes the approach or discourages it. This chemical negotiation, this molecular conversation between fungus and root, happens before any physical connection is made, before the first hypha touches the root surface. A conversation conducted entirely in the language of volatile chemistry, in the dark, in the soil, in silence. Once a mycorrhizal partnership is established, the communication continues. The fungus sends signals that tell the tree what nutrients it is providing. Phosphorus, arriving via the fungal network. Nitrogen, pulled from the soil. And the tree responds with signals that regulate how much sugar it sends in return. The exchange is not simply mechanical, not simply pipes carrying resources in both directions. It is negotiated, adjusted, regulated by a continuous back and forth of chemical information. If the tree has enough phosphorus, it signals the fungus to reduce its phosphorus delivery. If the fungus is receiving less sugar than it needs, it adjusts the rate at which it provides nutrients. The partnership is self-regulating, responsive, maintained by communication, by the continuous, invisible exchange of molecular information between partners. When a plant is attacked, the fungal network becomes an alarm system. A tree under assault by leaf-eating insects releases chemical stress signals. These signals enter the mycorrhizal network, travel through the hyphae, and reach the roots of neighboring trees. The neighboring trees, receiving these molecular warnings, respond. They begin producing defensive compounds, tannins, terpenes, compounds that make their leaves less palatable, less digestible. Before the insects have arrived, before any direct contact has been made, the warning has traveled through the network, through the fungal threads, from tree to tree. A silent alarm passed underground. The forest preparing its defenses before the threat has reached it. Fungi also communicate with each other. When two hyphae from different fungal individuals of the same species meet in the soil, something remarkable can happen. They may fuse, merge their cytoplasm, share their genetic material. But before this fusion occurs, before this most intimate of biological unions, the two hyphae communicate. Chemical signals are exchanged. Compatibility is assessed. Only if the signals are right, only if the chemistry says yes, does the fusion proceed. This is fungal courtship, conducted in chemistry at the microscopic scale, in the dark soil, when the answer is no, when the chemical signals indicate incompatibility, when two hyphae meet and the chemistry says these two individuals should not merge. The hyphae withdraw, turn away, grow in different directions. The boundary between incompatible fungal individuals is maintained by chemistry, by signals that say, this is mine, that is yours, the border is here. Fungi also use chemical signals in a process called quorum sensing, a term borrowed from the study of bacteria, where it was first discovered. Quorum sensing is the ability of a microbial population to detect its own density, to sense how many individuals of its own kind are present in a given space, and to change its behavior when a critical threshold, a quorum, is reached. In fungi, quorum sensing regulates important decisions. When a fungal population becomes dense enough, quorum sensing molecules accumulate in the environment. Their concentration crosses the threshold, and the population shifts its behavior. It may begin producing spores or forming different types of structures, or producing defensive compounds, or changing the way it interacts with its host. The individual fungus has no way of knowing how many other fungi are around it. But the chemistry of the environment tells it. The accumulated signals of all those individual organisms, building up in the soil water, reaching the threshold, triggering the collective response, a democracy of molecules, a census taken in chemistry. And then there are the electrical signals, perhaps the most surprising discovery in recent fungal science. In 2022, a researcher named Andrew Adamansky published observations of electrical signals propagating through fungal mycelium, spikes of electrical potential traveling along the hyphae at measurable speeds in patterns that, he noted, bore a structural resemblance to the action potentials of neurons. The electrical signals of a nervous system. This observation was extraordinary enough to generate significant scientific debate. Some researchers were cautious, noting that the electrical spikes might be byproducts of other cellular processes rather than true communication. But others found the patterns intriguing. The signals seemed to travel further and faster when the mycelium encountered food or was disturbed or came into contact with another organism, as if the network were reporting, alerting distant parts of itself, coordinating a response. Whether these electrical patterns constitute communication in any meaningful sense is still being determined, but the signals are real. The propagation is real, and the mycelium, threaded through kilometers of soil beneath the forest floor, is carrying them right now, tonight. There is a beautiful thought to hold as you rest. The forest floor is full of languages you will never learn. Chemical languages, molecular conversations conducted in the dark between organisms that have no brains, no intention, no awareness, and yet the conversations produce outcomes, alliances formed, defenses raised, nutrients shared, borders maintained, partnerships begun and sustained and adjusted, and sometimes ended. A whole world of relationship conducted in the only language available: chemistry, touch, electrical pulse. The ancient, wordless, continuous conversation of living things. The forest has been talking since before there were words, since before there were ears to hear. chemical signals drifting through soil and air tonight are older than language, older than thought. They do not need to be understood to do their work. They simply drift. They find their receptors. They trigger their responses. And the forest continues, as it has always continued, in quiet, wordless, chemical conversation. Let that conversation surround you now, the signals moving through the air you breathe, through the soil beneath the house, through the networks of every plant and tree and fungus in the world outside this room. A conversation you are part of, simply by breathing, simply by being alive, simply by resting here, in the dark, in the quiet, at the still center of a world that is always softly talking. We are almost at the end of our night together. Almost, but not quite yet. There is no hurry. There is never any hurry here. Let the night hold a little longer. Let the forest keep you for a few more quiet minutes. You have come a long way tonight. You have traveled deep into the world of the fungi, and it is worth, before the last of the night fades, taking a moment to simply rest in everything we have found. We began at the forest floor with the leaf litter, with the smell of damp soil, with the first pale threads of mycelium barely visible beneath a fallen log. And from there, we went deeper into the wood wide web, into the mycorrhizal networks threading between the roots of the oldest trees, the partnerships that sustain forests, the slow, patient exchange of nutrients and sugars between trees and fungi that has been running uninterrupted for hundreds of millions of years. We learned that the trees talk to each other through these networks, that a mother tree sends carbon to her seedlings in the dark, that a warning of insect attack travels underground from trunk to trunk, that the forest is not a collection of individuals competing for light, but a community, connected, communicating, sustained by the fungi that thread between them. We went into the history of the earth. 1 billion years of fungal patience, the spores in the ancient rock, the Rhynie chert, preserving the partnerships of 400 million years ago in perfect, microscopic detail. strange columns of prototaxites rising 8 meters from a Devonian shore, the first mycorrhizal threads extending from the first land plants into bare rock, making soil where there had only been stone, making the world habitable. One slow hyphal tip at a time. We sat with the decomposers, those quiet, essential fungi that return the fallen to the living. The white rot and the brown rot. The bracket fungi, slowly transforming the heartwood of a fallen oak. The ghost of the mycelium in the composting leaf, releasing the minerals of summer back into the soil of spring. Everything that falls, rising again, in a different form, in a different body, but present, still part of the great exchange. We found healing in the forest. Rise he on the old hardwood, chaga on the birch, turkey tail fanning from a fallen log, lion's mane cascading white and extraordinary from the wound of an old tree, and the accidental miracle of a September morning in London. A mold on a forgotten petri dish, a circle of dead bacteria, and the beginning of something that would save 200 million lives, all from a spore, drifting in through an open window. We followed the truffle into the soil. We breathed the petrichor. We traced the geosmin back to its source in the ancient bacteria of the earth. We stood in the winter forest and found the velvet shank fruiting through frost. We knelt at the edge of a cold stream and watched the star-shaped spores of the aquatic hyphomycetes drift in the current. We lay on the forest floor in the darkest hours and listened to the chemical conversation of the mycelium, the quorum sensing, the electrical pulses, the vocabulary of an ancient and wordless world. And now the night is almost over. The darkest hours are behind you. Somewhere very far away, the first suggestion of light is forming at the edge of the sky. Not yet visible, not yet felt, but building, the way everything in the forest builds, slowly, without announcement. The mycelium beneath the forest floor is still working. It worked all through the night. It worked while you slept. It will work through the morning, through the afternoon, through every season that comes, patient and continuous entirely without rest. That is what it means to be mycelium, to be always in motion, always reaching, always connecting, always transforming the world, one slow hyphal tip at a time. You have rested tonight in the company of the oldest kingdom on earth, the kingdom that came before the plants, before the animals, before the great forests and the flowering meadows and the coral reefs and the grasslands. The fungi were here first. They prepared the ground. They made the soil. They formed the partnerships that allowed